So are you going to give an introduction, Dr. Tomasi?
Yes, Professor Kathleen, yes, I will be the one in charge of introduce the session, the day, and of course all of you speakers and invited speakers. I will take care of it. No problem. No problem. So, okay, we are right on time. Good morning to all of you, to all the participants, the attendants, and I'm Stefano Tomasi. I am the chair of this morning session, and welcome, of course, to the second day of presentations of the autumn meeting for young chemists in biomedical fields. Today is the medicinal chemistry session, and this meeting has been organized by the young researchers from the Department of Pharmacy of the University of Naples Federico II, and has been conceived for acknowledged researchers, young researchers, postdocs, PhD students to share their science, to get in touch with each other, and of course to enlarge their knowledge. Before I start, I would like to thank our institutions and of course the Department of Pharmacy of the University of Naples Federico II, the Italian Chemical Society, our sponsors, Wiley Europe Chemistry and Elsevier, our invited speakers, and of course all of the attendants for their contributions that lay on the basis of this meeting, of this session. Just a brief information regarding the day, our session. Today we will have one plenary lecture by Professor Amedeo from the University of Zurich that I see already on the screen, ready to talk. We will have two keynotes, one in the morning by Dr. Neil London from the Weizmann Institute of Science. And in the afternoon, we will have another invited speaker, that is Professor Angela Russell from the University of Oxford. Overall, we will have 12 oral communication and 6 short communication. And this session will be divided into 2 sessions, one in the morning, one in the afternoon. So I think I've said everything basically. Now it's the turn for me to introduce Professor Amedeo Kaplish. I'm honored for this, to do this. Professor Amedeo Kaplish studied theoretical physics at ETH of Zurich, where he got his PhD in biomedical engineering and medical informatics. He then joined as postdoctoral fellow the laboratories of Martin Karplus at the Harvard University in the US, and then came back to the University of Zurich, where he is currently full professor in computational structural biology since 2001. His research interest covers very broad fields of biomedical sciences. It's very hard to summarize that, ranging from protein folding and stability, development of computational methodologies, and their application to the design of chemical probes and drug candidates. For me, I'm honored to introduce you, Professor Amedeo Kaplish. We are eager to listen to your lecture. And now I leave to you the stage, so feel free to start whenever you want, and welcome to the AMYC 2021. Please.
Oh, can you hear me?
Yes, yes, we can hear you. We see also your screen.
Okay.
I think it's in a full mode screen.
Yeah, very good. So it's a pleasure and an honor, particularly to speak in front of an audience of young chemists. I find it very exciting, and I think it's one of our duties, old and established professors, to really motivate the young generation. So this is a story on the medicinal chemistry of optimization of an inhibitor of an enzyme that is involved in epitranscriptomics. I'll explain you later on the details. It has anti-cancer potential. Most of this work was done by Aymeric Dolbois, a very talented French chemist who worked as a postdoc in my lab for 2 years and did a great medicinal chemistry optimization. So I have one slide of introduction because, as Dr. Tomasi said, in the past we have done lots of computational biology, molecular dynamic simulations, docking. In this slide, you see that we still do lots of docking, and actually our program for docking is very efficient. It takes around 1 second per fragment. And so you don't need a supercomputer, you just need a laptop or a desktop. And what is important then is to validate by biophysical and biochemical assays the docking predictions. We use many techniques like isothermal titration calorimetry, first resonance energy transfer, which is faster, more high throughput than calorimetry. And at the end, we use also a lot, or not only at the end, during the process, we use lots of protein crystallography to get the details of the interactions between our compounds and the protein of interest. Actually, this is one of our metal 3 inhibitors, and you can realize here the 2 nice hydrogen bonds of the cyclic lactam with the protein target. So the story of today is essentially partitioned like a paper because it's mainly the content of one paper which we published a couple of months ago on JMET Chem. I'll give you the reference towards the end. And actually, I will be quite short on the objectives and biological investigations. I will try to focus on the medicinal chemistry optimization, which was supported by protein X-ray crystallography. So as I briefly mentioned at the beginning, we are interested in RNA modifying enzymes, but let's start with the dogma of molecular biology. DNA transcripted into RNA and then translated into protein. But there are mechanisms beyond this dogma, and there are that regulate gene expressions at the level of modification of DNA, so-called epigenetics, and modifications of RNA, so-called epitranscriptomics. And today we focus on RNA modifications, which there are plenty in all organisms, actually. More than 100, probably more than 150 reported RNA nucleoside modifications. There are some examples on these slides on uridine, cytidine, guanosine. Here notice, for instance, M7 guanosine, which modifies the charge. So the formal charge of guanosine on the nucleic acid base is 0. Once you add a methyl group here, it actually has a +1 charge, which is a quite substantial change. In adenosine, we will focus today on M6A, so a methylation at nitrogen, exocyclic nitrogen 6 on adenosine, which is the most frequent modification of RNA. So it's the most abundant. It's in about 25% of all cellular messenger RNA, but it's also identified in ribosomal and micro RNA. There is experimental evidence for the involvement of this modification of this simple methyl group, so substitution of an hydrogen by a CH3, involvement in circadian clock regulation, RNA stability, cell differentiation, DNA damage response, and of course, all of this has potential implications in cancer. And in fact, there are links to numerous diseases, not only hematological malignancies like leukemia, but also solid tumors like breast, prostate, and others. There is also potential involvement in virus infection because of M6A modifications in viral RNA. So this might become also even more interesting if one considers that SARS coronavirus 2 also has M6A modifications on RNA. So back to the modifications, there are 3 types of protein involved in producing the modification, recognizing it. The first is the writer enzyme, which adds the methyl group to the adenine. Then you have readers enzyme that recognize the modification, and erasers that make the modification reversible. We focus today on the writer enzyme, the so-called methyl-3, methyl-14 complex. It's an heterodimer. I'll show you later on the crystal structure. And it's the main player of M6A methylation. It does need a co-substrate, like most methylations of proteins and nucleic acids by enzymes. The co-substrate is S-adenosyl methionine here, which is the donor of the methyl group. So I told you that it's a dimeric structure. It consists of 2 protein chains, chain methyl-3, chain methyl-14. By the way, methyl stays for methyltransferase-like protein. They have these 2 chains and folds have similar structure but different functions. There is biochemical evidence that methyl-3 is the real catalytic subunit, because if you truncate methyl-3, the activity drops dramatically, whereas you can truncate methyl-14 and the activity is still high, in the presence of some, of course, of the cofactor S-adenosyl methionine. So methyl-3, to summarize, methyl-3 is the catalytic core, methyl-14 is a scaffold that helps binding the real substrate RNA. This is the crystal structures. 3 groups solved the crystal structure in 2016, methyl-3, methyl-14, the interface, which might be essential, which is essential for binding RNA. It has a charged groove, positively charged groove. And then this is the binding pocket of SAM, which is the region that we targeted at the beginning by docking. Actually, already we started this project about 4 years ago. When we started, there were only 2 inhibitors. Actually, we produced one of these 2 inhibitors in the first 2 years. So again, we want to compete SAM, and one of the 2 known inhibitors when AMYC started actually was our own. This compound here, UZH for University of Zurich 1, had a decent inhibitory activity. So 280 nanomolar measured by our own first resonance energy transfer assay with recombinant enzyme in solution. More recently, actually, 5 months ago, a company called Storm published a low nanomolar inhibitor, which was not actually known when we started this study. So the point is that there are essentially only these 2 compounds. Their ADME, absorption, distribution, metabolism, excretion activities were not studied that much, neither by us nor by others. And there was limited availability of this compound, actually, particularly of our, now not anymore, because actually we recently realized that there is a company that sells our UZH1 compound. One can buy it, actually. Many groups requested this compound from us. We shipped it to many groups, but then later on we realized that the company makes it. I don't make any profit out of it, but if you're interested in our first chemical probe, it's commercially available. Most probably the most potent compound that is the content of this story will also be probably sold by 1 or 2 companies in the future. So before I go to the medicinal chemistry optimization, I would like to mention that it is very important to have an assay, a biochem or biophysical assay, to measure inhibitory activity. So you have to have an assay. If you want to do a real medicinal chemistry optimization, you have to have an assay that is efficient and reliable. And we decided actually to develop our own assay about 4 years ago, because there were already roughly 3 different methods to measure inhibitory activity of metal 3 and metal 14, but they had drawbacks. So mass spectrometry, one was based on mass spectrometry, another radioactivity, the third one was based on ELISA. Of course, the mass spectrometry one is low throughput, which is a main drawback. It has other disadvantages. The isolation can be laborious, it's time consuming for us, it needs deuterated sum. The radioactivity based is not really interesting to us. It needs deuterated sum, special equipment, it's cumbersome. And ELISA, well, you know about ELISA, it's not very quantitative. You need a specific antibody. It's actually requires several washing steps. So we thought that a new assay was desired, and we developed our own assay based on the reader. So this was a brilliant idea of a PhD student of mine, Lars Widmer. We published actually the method he established 2 years ago in ACS analytical chemistry. So he had the brilliant idea of using the natural reader to recognize the modification. So in a simplified scheme, you have here the substrate with the FRET acceptor and the natural reader protein with the FRET donor, and there is no interaction if adenine is not modified because the reader selectively binds to the methylated adenine. So there is no FRET. Once you add the enzyme methyl 3, which catalyzes the reaction, the adenine is now modified into M6A, and the reader recognizes it so that one has FRET efficiency transfer. So again, the brilliant idea to use the natural reader, which is specific for the methylated adenine. And so instead of using antibodies that tend to be difficult and not specific. The demethylase activity is also measurable, and it is high throughput. It does need low substrate enzyme consumption, no washing step, no specific equipment. We have measured, I don't know, nearly 100 compounds with in the past months with this assay, and it works very reliably. Right, so now I go to the objective of the study. So we had as a starting point compound 1, which was similar to the UZH1 I showed you. It was one of our early hit obtained by docking, 7 micromolar in our time-resolved FRET assay. It's not great, and it's a complex compound with one stereogenic center, quite flexible linker region. We knew its pose. It was a SAM competitor. We solved the crystal structure of this heat compound 1. We saw that it had a couple of hydrogen bonds. We also realized that this carbonyl group here is not involved in any hydrogen bond. And so this was very important to further improve into a lead compound, which was our aim. Our aim was a potency below 50 nanomolar. You will see later on that we reached actually 5 nanomolar, which is 10 times better than our original goal. We also, of course, wanted a compound that is selective against other RNA methyltransferases and, of course, protein methyltransferases. And then good ADME properties for biological investigations. So Aymeric, the postdoc, decomposed the compound in 3 parts: the head, the body, and the tail. So the original heat, 7 micromolar heat, was decomposed into these 3 parts. And we had already optimized the tail here during the development or during the optimization of UZH1. So you will see that the next slides will focus on optimization of the body or of the central part of the molecule, and a bit on the optimization of the head. So the body optimization, again, started from compound 1, which was a low micromolar compound with a relatively high molecular weight, which resulted in a relatively mediocre ligand efficiency. I'll explain you later on the ligand efficiency. In any case, we realized from the crystal structure that we could simplify the initial heat compound by removing the stereogenic center. In other words, we thought to shift the linkage point from here to this carbon atom. And also we realized that this carbonyl here was not involved in the protein. So Aymeric decided to simplify, and he synthesized compound 2 which now does not have the stereogenic genter anymore, and it does not have the carbonyl. These had similar potency, but much or substantially lower molecular weight, so that its ligand efficiency increased and no chiral center. So lower molecular weight, better ligand efficiency, which is the measured binding affinity divided by the number of heavy atoms. So it's a measure of how good each atom of the ligand interacts with the protein. We also did 2 pyridine derivatives, which had again similar potency, but better predicted lipophilic ligand efficiency, which is the difference between the potency and the calculated log P. Now, so this was the very important first step, simplify the molecule. In other words, removing the stereogenic center and removing, this amide, or removing the carbonyl of the amide, which is not involved in hydrogen bonds. So the synthesis, I will not go into the details of the synthesis briefly here. It started with an alkylation of the 4,4-dimethyl piperidine. Then there was Buchwald-Hartwig amination, which afforded this compound upon Bock deprotection. Originally, this group here at the Bock deprotected here. And then by aromatic nucleophilic substitution with 4,6-dichloropyrimidine, we could obtain this compound. And then again, an SNR reaction with methylamino, which afforded the final compound too. The yields are quite decent for medicinal chemistry optimization. So 99% here, of course, and 33% here on the last step, and 36% here over this 1, 2, and the SNR step. Right. But the potency was not great, so we need to improve the potency. And the first thing we did was adding a benzyl moiety, which improved the potency by a factor of about 7. And we rationalized that by a crystal structure. The benzyl ring here makes pi stacking with the guanidinium of an arginine. But we still had the problem of flexibility. And so Aymeric thought about cyclizing this benzyl derivative. And this was the first Piro compound he made, which improved the potency substantially by a factor of about 3. And the reason is because the compound has a certain entropy, which is frozen upon binding, and with the Spiro rigidification, there is a minor entropic penalty. We solved the crystal structure, and again, the design of Aymeric corresponded to reality. The crystal structure validated his design. So with Aymeric thought the cyclization is a good strategy, but there was a loss of the hydrogen bond to glycine 550 here, because of course this was a donor group, an hydroxyl OH donating to the glycine 550, and there is no donor group on the spiral. So we thought to reintroduce the hydrogen bond to glutamine 550. And actually, Aymeric thought about the lactam, so he synthesized a Spiro compound with the lactam here, which had a substantial improvement. Now we are in the 2-digit nanomolar range, 37 nanomolar, good ligand efficiency. So this was a significant potency improvement. And when we solved the crystal structure, it was very gratifying that the design of Aymeric actually was correct. We captured 2 additional hydrogen bonds, or the lactam NH and CO group is involved in a double hydrogen bond with the glutamine 550 side chains, which explains, at least in part, this significant improvement in affinity. I have a slide here on the detail of the synthesis of the Spiro group, which actually was Bock protected, as I told you earlier, but I don't want to go into the details here because of time. Right, the synthesis of compound 7, on the other hand, was very similar as the previous synthesis. So again here, alkylation, then Buchwald-Hartwig, which afforded this linkage here with the secondary or tertiary amino here on the spiral. This was Mach protected. It got deprotected here in this step 2, and then aromatic nucleophilic substitution with 4,6-dichloropyrimidine, and then another SNR with benzyl amino to afford compound C7 in quite good yield. So the potency was good now, was double-digit nanomolar. Now let's think about also about other properties, physicochemical and ADME. When we measured actually permeability, it was bad. So you will see here two numbers on this permeability data. The first number is the real permeability going from outside to inside of the membrane of the cell passing through the membrane, and the second number is the efflux. So it's the ratio between how much of the compound gets in and how much it gets out. So in principle, you want the first number to be very high and the second number to be low. You want the ratio to be low. In this case, it was really poor because it had a low permeability and high efflux. Also, the rat liver metabolic stability was not good because it was only 8 minutes, the tau 1/2. So the half time was 8 minutes, which is not enough. It should be at least 20, 30 minutes, preferably more. And so the first thing we tried was to remove the nitrogen, and this actually increased permeability. Now you have much better permeability and much lower efflux, but the metabolic stability was still quite low. So we removed the benzyl, and we got a 90 nanomolar compound, which is actually not bad, which has very good solubility, kinetic solubility, very good rat liver metabolic stability, but again, poor permeability, sorry. So again, so very low permeability, very high efflux ratio. So it means it's pumped out, it's not good. So then we thought about other spiro group. Can we modify this part of the molecule to improve the ADME? So this was a spiroacetidine here, and then a spirourea. You can recognize the urea group here. And then we also did a negative control, methylating the nitrogen. We knew that the nitrogen was donating a hydrogen bond, and so we wanted to do also negative control to see what happened. And in fact, the potency of these three compounds is poor. High potency loss. And so we went ahead and we thought, well, let's try to improve the permeability by optimizing the head. As I told you at the beginning, Aymeric had divided the compound into head, body, and tail, and now it was the turn of the head. And so we did alkyl group modification here, and the dimethyl derivative had a poorer potency, so we lost a factor of 10, so we didn't measure here solubility and permeability. This compound here with the propyl group here was also not great, whereas this compound with the cyclopropyl was good, and that's why we measured also the ADME properties, so very good solubility, very good metabolic stability, but this is not great. Again, there is too much efflux. So this were not really interesting improvement. This might be interesting with the cyclopropyl, but the permeability is not perfect. So permeability improvement, cyclization, these were other attempts we tried, or Aymeric tried, by cyclizing here the compound in the head. The potency was okay, but there was a slight decrease in the ADME properties. We made this compound here without the nitrogen here, a poor potency, too much loss. We know that this hydrogen, both these two hydrogens are involved in hydrogen bonds with the protein, so it was some sort of negative control as well. With the chlorine, things went, the potency is good, but solubility didn't improve, actually deteriorated. And metabolic stability is okay, but not great. So we decided to stick with a single methyl here at NH, and we decided to turn our attention now to the phenyl ring, which we had modified in the past only by modifying into a pyridine. So Aymeric thought to add the fluorine, which is done frequently in medicinal chemistry optimization, and we got a promising improvement by a factor of about 2.5 in inhibitory activity, and this was very interesting now. So the permeability was substantially better, 4 times better, and the efflux decreased. So the compound gets in and stays. So we tried fluorine in the other position. So this was here. This is the other position for the fluorine. But in this case, the permeability is actually poor. And then the idea was to, we saw the crystal structures of both. We saw certain interesting interactions. This fluorine here was making an interaction with the pi system of the amide of the proline. This fluorine in compound 21 was making interactions with a couple of aliphatic and aromatic rings, aliphatic group and aromatic ring. And so we decided why not to combine them. And make what is now our lead compound, added both fluorine. So now we got a compound with adequate properties, 5 nanomolar IC50, good solubility, very good permeability, low efflux ratio, and the rat liver metabolic stability, the tau 1/2 is about 25 minutes, which is okay. It's not great, but it's acceptable, particularly for drugs against cancer. So this is now our lead compound. We called it UZH2, University of Zurich 2, because we already had UZH1. So it has improved substantially because UZH1 was high nanomolar, and we don't have the stereocenter anymore. It's more rigid and has very good properties. So again, the synthesis is very similar, so there is no need to go into the details. Again, alkylation, and then Buchwald-Hartwig, the protection, and then the first SNR, and the second SNR here with methylamino, which affords compound 22 in quite decent yields. So now on the selectivity, this is a very important point which I have not mentioned up to now. You want to have a compound that blocks your target protein, but not other cognate enzymes. So we knew that methyl 314 is the major M6A methyltransferase, but there are plenty of other RNA and DNA methyltransferases and protein methyltransferases. So we expressed and then purified in my group methyl 16, which is another M6A methyltransferase, and methyl 1, which is an M7 guanosine methyltransferase. And we carry out the thermal shift assay using SAH, so the co-product as positive control. The thermal shift assay measures the stability of the protein, the thermal stability of the protein, which should increase in the presence of an inhibitor or compound that stabilizes the native fold. And you will see that on the target, our compound shifts, so increase the melting temperature, which is good. It means that it binds potently to the target. The same for SAH, though the concentration here is quite different. So our compound at tenfold lower concentration has a larger shift than the co-product. Whereas our compound, UZH2, does not shift, so does not bind to methyl 16 and MET1, which is a good evidence of selectivity. We also have to provide evidence that the compound binds to the target in a cellular environment. So for that, we use the so-called in-cell pulse, which uses target methyl 3 in these cells, in these cell types, and uses heat to see if compound binds, and there is protein stabilization. And TAC plus enzyme, so it's a luminescence assay, there is light emission, whereas if the compound does not bind, the protein is not stabilized, it denaturates, and so there is reduced light. And when we measured our UZH2 in HEK cells by the in-cell pulse assay, we obtained effective concentration 50, so a concentration at which you get 50% of response of 2 micromolar. So one thing I have to explain is that there is a factor of about 500 to 1,000 deterioration from an IC50 of 5 nanomolar to a cellular EC50 of 2 micromolar. And the reason is because the cell has a very high concentration of SAM, of the co-substrate. SAM has a concentration between 20 and 50 micromolar in the cell, depending on the cell type, and so competes substantially so that cellular value of low micromolar are very good. When we did the SETSA, the cellular thermal shift assay, which is similar to this one, but not identical, on another type of cell, these are leukemia now cell lines, we got actually an EC50 of 850 nanomolar, which is actually very interesting. This is a sub-micro, high nanomolar, but sub-micro. Again, because of the concentration of SAM in the cell is much higher than when one does a biochemical assay. So this is proof of cellular target engagement. Then we did the most important experiment, which is using mass spectrometry, MSMS, to determine the cellular level of M6A, or better, the reduction of M6A upon inhibition of metal 314. So we measure the M6A over A ratio in messenger RNA, and on two different cell lines, a leukemia cell line, a prostate cancer cell line, and we obtained EC50 in the low micromolar, actually even sub-micromolar range. So this is the direct demonstration that the compound reduces the amount of M6A in the cell. So direct readout. These are the details of the experiment. Again, it's done by mass spectrometry. So the bottom line here, sub-micromolar concentration are enough for decreasing substantially M6A level in these leukemia cell lines. On prostate cancer cell lines, PC3, the EC50 is quite well. So it seems that there is a different heterogeneous involvement of metal 3 in different cancer cell lines. Then we wanted to be sure that within the cell we were not hitting other RNA methyltransferases. And so we measured, again, not only in messenger RNA, but also in total RNA, not only M6A over A, but also the ratio of M1A over A, M6AM, where here there is a methyl also on the ribose, and M67G. So our compound UZH2 is high selective for M6A in messenger RNA, which means that it's high selective for metal 3 and does not hit other methyltransferases that are responsible for other either non-M6A RNA modifications or modifications M6A, but in ribosomal or transfer RNA and not in messenger RNA. Right, so I am close to the end. So one slide of conclusions. We have now a potent metal 3 inhibitor called UZH2, which is a 5 nanomolar compound. It has good ADME properties. We have shown evidence of selectivity against related proteins like methyltransferase like 16 and methyltransferase like 1. We have strong evidence of cellular target engagement and good ADME properties. The liver metabolic stability might still has to be improved, but 24 minutes as half-time is already good enough for cancer applications. And the most important thing is the previous slide I showed you that we see M6A level reduction in cancer cells. So all of this was published actually a couple of months ago on Journal of Medicinal Chemistry with this long title here because this scaffold here is a 1493 azaspiro undecan to one to 1, and as I said, it's work mainly done by the chemist Dogma. And we started from a 7 micromolar hit, and we obtained a 5 nanomolar lead, which is a 1,400-fold potency improvement, which was obtained in about 1.5 years. But importantly, it was supported and in part also guided by protein crystallography. So solving crystal structures of intermediate compounds with the target. So if you want, this is protein crystallography based medicinal chemistry optimization. And yeah, I am already at the acknowledgements. The people who did the work, again, Aymeric here, the chemist. Rajiv Bedi is a PhD student who has solved nearly 50 crystal structures of metal 3, metal 14 in the complex with our inhibitors. 45 of them have already deposited and released in the protein data bank. He has solved also structures of reader domains. This is the group funding not only from the University of Zurich, but also, of course, from the Swiss National Science Foundation and the Cancer Society of Switzerland. And thank you very much for your attention. I would be glad to have many questions.
Okay, Professor Keflich, thank you for your clear presentation, full of informations, characterizations, and thank you for bringing us into the epitranscriptome world again, I would say. This is a very new target, and yes, let me see. Of course, congratulations for your work and research. Let me see if there are some questions in our session. I guess, yes, there is one question from Margherita Brindisi.
Yes, good morning, Professor Keflich. Can you hear me?
Yes.
Okay, so thank you first of all for the nice presentation, because it's a very, very nice example of structure-based design. Related to this, it's also the fact that you are really showing in your molecules how is it important also to increase the fraction of sp3 hybridized carbon, because all these spiral moieties are really interesting and also are very important for chemical-physical properties. Are you thinking also of modifying, in this sense, the defluorinated aromatic ring? Are you thinking to aliphatic moieties or something for increasing sp3 fraction?
Yeah, thank you. This is an important, interesting question. I don't think we have thought about that. I mean, we have tried other substituents. You can imagine we tried other substituents. And in fact, we have another couple of extremely potent compounds with other substituents on that phenyl ring. But I think we could also think about changing the ring completely. And I mean, I don't know, we would have to think very carefully about the synthesis. But yeah, replacing that phenyl with a ring that has at least one sp3 carbon might be interesting.
Yeah, I'm referring for example, to bicyclopentanes, which are classical phenyl isoster, this kind of.
Yeah, I mean, thank you very much. This is a good suggestion. I will discuss it with my chemist. I mean, Aymeric has left, but we have another chemist, and so yeah, I will discuss it. Thank you.
Thank you.
Professor Keflich, I don't know if there are other questions. Yes, there is another one. There's another one from Dr. Francesco Fiorentino from the University of Rome Sapienza. This is one of our next speaker in the afternoon. He's asking to you, okay, he congratulates with you, and he's wondering whether you have evaluated the selectivity of your best inhibitor, I guess you use at H2, over a panel of other SAM-dependent methyltransferases, such as histone-methyltransferases.
Yeah, this is also an important question. Thank you for asking it. Yes, we did evaluate, but you know, histone are peptides, and so there is no affinity at all. The binding site is quite different. Even the SAM pocket is quite different between protein methyltransferases and RNA methyltransferases. But so we did that. We did that with the UZH1 and also with UZH2, and there is no binding to histone methyltransferases and other protein methyltransferases in general. You know, I didn't report this because it is more stringent to show selectivity against other RNA methyltransferases. But we of course also did the protein methyltransferases.
Okay, so I think there aren't any other questions. So I would like to take the chance to make one question of mine. I have also have lots of questions, but I will restrict the selection. The first one, it's a very broad question, I would say, and it is if you have considered in the future maybe to develop some PPI, protein-protein interactions inhibitors, disruptors between metal 3 and 14 that are heterodimer. I've seen it from the crystal structure. So
very good point. Yeah, we are thinking about that. I cannot say more, but it's of course a very good point, because it would also bring selectivity, because that interface is unique to metal 3, metal 14, and yeah, I mean we have done docking. We have not invested substantial resources in chemistry on optimizing compounds for the interface, because we have limited resources. But if any of you is interested in developing, in further developing fragments that hit the interface, you should contact me. And
okay.
I have limited chemistry resources.
Oh, okay, so whoever is listening to, you can take the chance, me included, I would say. Yes. So I think I would have also other questions, but because of time issues, I think I have to stop myself. So thank you once again for being with us, for sharing your big results, and thanks for your time once again. And
well, thank you, Dr. Tomasi, for the opportunity and for inviting me. And as I said earlier, it is a real pleasure and an honor to talk in front of an audience of young chemists. Thank you.
That was our purpose. Thank you to you too. So once again, bye, and hope to see you in the next edition. Thank you very much. Bye. So now it's time to move on. I see already Dr. Neil London on our screen. Once again, welcome. So I would like to spend some words about Dr. London. I'm very honored for that. So welcome once again. Dr. London completed his PhD in computational structural biology at the Hebrew University in 2012. He then moved to the University of California at San Francisco as a postdoctoral fellow. And in 2015, he joined the Weizmann Institute of Science as assistant professor at the Department of Chemical and Structural Biology. Dr. London works in the field of chemical biology and drug discovery, and is involved in development of new technologies applied to covalent inhibitors. Dr. London also was recently awarded the eminent EFMC, European Federation of Medicinal Chemistry Prize for Younger Investigator in Academia. So congratulations for this acknowledgement, for this prize. And so Dr. London will present his work regarding the entitled Teaching the Acrylamides New Tricks. So once again, welcome, and we are eager to listen to your presentation, and feel free to start whenever you want to.
Thank you very much, Stefano, for the kind introduction and for the invitation to speak in this exciting meeting. And so, as Stefano mentioned, our lab is focused on covalent ligand discovery. As some of you may know, compounds that are able to form a covalent bond with their target protein has several advantages, both for chemical biology and for drug discovery, over their counterparts, which are non-covalent. These include increased potency, typically due to the formation of the covalent bond, a longer duration of action, since the protein needs to be resynthesized in order to regain its activity. And they can be very selective if you're targeting a non-conserved residue that's only found on your target protein. For these reasons, recently there were several FDA approval of rationally designed covalent drugs, and some clinical candidates are now in the pipeline for challenging targets, such as, for instance, KRAS G12C. Despite all these advantages, it's still pretty challenging to design and discover covalent inhibitors, and that's the focus of our lab. And over the years, we developed several technologies to facilitate that, so including DocCovalent, which is a covalent virtual screening platform for discovery of covalent inhibitors, a platform, excuse me, an empirical platform for covalent fragment screening, combining the power of covalency with the efficient exploration of chemical space by very low molecular weight compounds. A computational protocol called Covalentizer that derivatides known reversible binders to make them into irreversible binders based on their binding close to a target cysteine. We also reported covalent PROTACs, both reversible and irreversible for targeted protein degradation. And recently we described the covalent peptide docking algorithm, where if you have a peptide protein interaction and a cysteine on the receptor somewhere near the peptide, it can suggest which electrophilic amino acid you can incorporate into the peptide to turn it into an irreversible peptide binder. But today I want to talk to you about a new electrophile class we developed, and it's based on acrylamides. So acrylamides, which you can see here, are the most prevalent electrophiles in approved covalent inhibitors. This is because they balance very low reactivity, but they're still able to form the requisite covalent bond. Here, ibrutinib will be one of the stars of today's talk. You can see the acrylamide found here. And the knowledge in the field says that the intrinsic thiol reactivity of the beta carbon of acrylamide can be attenuated if you substitute either the alpha or the beta position. Typically, you lose the reactivity of acrylamides or substantially lower it. Whereas there are some examples where, for instance, if you install an electron with rowing group, such as a nitrile, at the alpha position, you can activate this position, but also makes the covalent bond reversible, so more labile. For this reason, we were surprised where in one of our campaigns, we made this compound with an alpha substitution of a dimethylamine. And upon incubation with the target protein, we got the formation of a covalent bond, but this dimethylamine group was not present in the complex. We did some literature search, and in the bioconjugation literature, we discovered that these alpha substituted substitute the alpha methacrylamides were actually reported to be able to react with thiols, and instead of undergoing the typical Michael addition, they undergo a conjugated addition elimination reaction, which at the end liberates this heterosubstitution as a leaving group while forming a covalent bond with the protein and reconstituting the acrylamide. We wanted to explore this chemistry, so we made this model compound in which there's a benzylamine as a model amine on which we installed the acrylamide, and this coumarine acts as a heterosubstitution at the alpha position. This can either go the conjugated addition elimination that we described or a typical Michael addition. And to follow the reaction, we used mass spectrometry. You can see after half an hour, we detect the starting material. But if we keep the reaction for 48 hours, we lose all the starting material, and we see an accumulation of coumarine and the GSH substitution, and it's this adduct rather than the Michael addition that we witness, so favoring the addition elimination reaction. If we follow the rate of elimination of the starting product and the rate of accumulation of coumarine, they're identical, suggesting it's a concomitant release of the coumarine upon binding of the glutathione to the acrylamide. To further explore the scope of this electrophile, based on this benzylamine, we made a small library of different heterosubstitutions, including amine-based substitutions and oxygen-based substitutions, and we measured the reactivity towards glutathione. We measured the T-half of glutathione. So you can see that the acrylamide is very slow to react with glutathione. The methylacrylamide is actually completely dead for this reaction, but installing the heterosubstitution actually increased the rate of the reaction with glutathione, and in almost all cases, it gave the substitution product rather than the addition product. Importantly, we see a very nice correlation between the reactivity, the glutathione reactivity of these compounds, and the pKa or pKb of the leaving group. So this gives us a dial or a handle with which we can basically attenuate the reactivity of this new electrophile class. We wanted to explore it in the context of targeted covalent inhibitors. For that, we took the scaffold of ibrutinib, this amine here, which we called IBRH. Ibrutinib has an acrylamide at this position. We installed various heterosubstitutions on the methacrylamide version, including just the methylacrylamide as a negative control. when we incubate these with BTK and follow the reaction by LCMS, you can see that almost all of the substitutions are as fast as ibrutinib, fully labeling the protein within 5 minutes. The only two exceptions are our negative control, which is much, much slower to react with BTK, and this compound here, the methylaniline, which reacts much slower as well. In vitro kinase activity assay, you can see a whole range of activities with a couple of the compounds actually being more potent than ibrutinib, which is in and of itself a 200 picomolar inhibitor in this assay, and we get about twice better compounds with some of the substitution, whereas other substitutions are 5-fold or even less potent. And again, the negative control gives us a 100-fold loss in activity in this assay. To show that this chemistry is also compatible with cells, this is a primary B cell assay where we activate B cells with anti-IgM, and we follow the expression of CD86 on the surface. And you can see that 4 of our derivatives gives very similar dose response curves to ibrutinib. So this chemistry also works in cells and not only in the test tube. We were surprised when we characterized the thiol reactivity of some of the ibrutinib derivatives to realize that we have derivatives such as 3C and 3F, which are less reactive than ibrutinib. If you remember from the model benzylamine compounds, they're typically more reactive than the acrylamide, but here they're less reactive than the acrylamide, but at the same time, they're also more potent in the in vitro kinase activity assay than ibrutinib. This would suggest they might be more selective in cells. To evaluate this hypothesis, we ran 2 chemoproteomic screens. In the first, we either incubated or not incubate cells with our inhibitors, then labeled the cells with isotopically labeled iodoacetamide alkyne, which in this sample would be competed by any off-target of our compounds. Then we mix the population and we measure the ratio of the different isotope of the iodoacetamide alkyne, and by this we can identify off targets of either ibrutinib or our compounds. You can see we identified about 40 off targets for ibrutinib and about 30 off targets for our compound. So ever so slightly improvement in selectivity, but nothing to write home about. But in this experiment, we didn't identify BTK itself, since it's lowly expressed and we're not specifically enriching for it. So we did a complementary chemoproteomics experiment where the competition now was ibrutinib alkyne, and again we competed off with our inhibitors or ibrutinib, and we used pull down to compare the enrichment in the competed and non-competed cells. And you can see here that we identify BTK as the most prominent target of both ibrutinib and our compounds, and we can identify a few of the off-targets. Again, we have about half as many off-target as ibrutinib, but the numbers are low, so it's still not super convincing. So to be extra convinced, we ran in vitro kinase activity assays on the 5 most prominent off-targets, kinase off-targets of ibrutinib, and you can see the results here. Pay attention to the orange and pink compounds, which you can see are much less potent against all of the 5 off targets, but are actually more potent than ibrutinib against BTK, the on target. And this is quantified in this table. And if you focus on the orange and pink compounds, you can see, for instance, for ITK, ibrutinib only has a 300-fold selectivity for BTK over ITK, and our compounds have a 3,600-fold selectivity. For ERB2, ibrutinib has only 40-fold selectivity over BTK, and we have 2,400 fold selectivity. So these compounds are in the same time more potent and more selective, which is not a simple thing to do when talking about an FDA-approved covalent inhibitor. But perhaps an even more exciting property of this chemistry is that you can functionalize covalent inhibitors by putting functional groups rather than just simple chemical modification. So here, when we put a coumarin at the alpha position, you can see that this compound 3K on its own is not fluorescent, BTK is not fluorescent, but upon mixture, you get very high fluorescence and very fast as well. If you pre-incubate with a reversible BTK inhibitor, the scaffold of ibrutinib, you can slow down the fluorescence formation by competing for the active site. And LCMS shows that the adduct after this reaction is indeed just the ibrutinib adduct without the coumarin. We can slow down the reaction by using lower concentration, and now we can actually see the initial formation of fluorescence. This is specific as BSA at 10 micromolar does not give any fluorescence signal, and we can block the reaction by iodoacetamide, which blocks the cysteine and then does not allow the fluorophore to be released. This is general. We showed the same turn-on fluorescence with an EGFR scaffold. This is a derivative of afatinib. You can see the turn-on fluorescence, and LCMS shows the right adduct mass for the remaining part of the compound. And for KRAS G12C, this is based on Amgen's compound that was recently approved. You can see turn-on fluorescence with KRAS and the correct adduct mass remaining on the protein. To test a different chemistry, instead of a fluorophore, we use this Chemi Luminophore reported by Doron Shabbat's group in several publications. Essentially, when this leaves as a phenolate, electrons move to the dioxetane, open the dioxetane, and releases a photon. So here you can see the compound is not luminescent in and of itself, neither is BTK, but upon mixing, we get a very high peak with very good signal-to-noise ratio of chemoluminescence, and we can block this chemoluminescence using the reversible ibrutinib amine. So this looks like a very nice assay for looking for potentially other inhibitors for BTK. So we ran a medium throughput screen with 3,500 bioactive molecules where we pre-incubated them and looked for chemiluminescence. And you can see that out of this library of bioactives, we have enrichment, maybe unsurprisingly, for kinase inhibitors. We're able to detect all of the known BTK inhibitors completely inhibiting the chemiluminescence, but many other compounds as well. And we followed up on 4 of these kinase inhibitors that were not previously annotated to be BTK inhibitors, but we can show that both in vitro and actually in cells, these are low nanomolar BTK inhibitors that we could identify with this simple assay system. So to summarize this part of the talk, we presented alpha-substituted methacrylamides as a new class of electrophiles that can show improved potency and selectivity, has a tunable reactivity, and importantly, we term this covalent ligand-directed release or Calder chemistry, enable us to functionalize this group. We showed fluorescence and luminescence, but you can also imagine turn-on toxicity or the release of a secondary inhibitor. And current investigations in cells and in vivo for these types of compounds are ongoing in our lab. For the second part of my talk, I'll switch the directionality of the acrylamide. So if for a Calder inhibitor, we talked about an inhibitor, an acrylamide, and a leaving group. If you just move the carbonyl, now the inhibitor itself can act as a leaving group, and this part of the molecule can stay as a tag covalently attached to the protein. So the compound will bind, the beta carbon stays in the exact same position for thiol attack by the target cysteine, but the addition elimination reaction will now eliminate the inhibitor, vacating the binding site and tagging the protein. We again used the scaffold of ibrutinib to make a series of potential tags with either alkynes for click chemistry labeling or various fluorophores or just amino acid derivatives to modify the cysteine residue. And in LCMS experiments, you can see that almost all of these compounds are able to fully label BTK within 10 minutes and certainly within 2 hours, whereas a few of the compounds are a little less reactive. For instance, this alkyne, if it's not connected through a secondary, through this primary amine, but through secondary amine, shows much reduced reactivity, which we can improve the selectivity of the compound through this modification of the chemistry. Again, we show generality by installing these tags. So the same tag here against either BTK again, but using a different inhibitor, evobrutinib instead of ibrutinib. And you see that the protein mass shifts by only this small tag and not the whole compound. KRAS using AMG 510. Again, same adduct formation. And this is an example of a PL-Pro covalent inhibitor from SARS-CoV-2 that we recently discovered, again, only leaving the tag and not the inhibitor. So If you take this alkyne derivative and you put it on cells, you can see that, and then we run the lysate gel and click them with fluorophore. You see that at 500 nanomolar, there are many off targets, but when you lower the concentration to 10 nanomolar, you see only 4 bands. This band is BTK, this band is BLK, a prominent off target of ibrutinib, and we basically get only 2 off targets. As I said, we can improve the selectivity by changing the chemistry here. So now you see only 3 bands, so only the 30 kilo downturn remains. These 2 bands are competed by ibrutinib. So, you know, they're off targets of the scaffold and not the colder chemistry. And this is basically the only new off target for this probe. We use chemoproteomics to identify this band. So BLK and BTK were indeed recognized, and MCAT and ADK are these 2 additional bands. They're both very abundant proteins, which may explain why we see this somewhat additional labeling. Maybe most important for this tagging strategy is that we leave the enzyme that we're tagging intact and active, since the inhibitors leaves the binding site. So here you can see that we can activate BTK autophosphorylation with anti-IgM. Ibrutinib is able to inhibit this this activation, but because it's reversible, if you wash, you're able to regain activity. Ibrutinib completely blocks the phosphorylation and is resistant to washing since it's irreversible. But 2 of our probes actually retain the activity both with or without washing. So tagging the protein, as I just showed you in the gels, but also retaining the activity. To make extra sure that this activity that we're seeing here is not due to unlabeled BTK, so if our labeling is incomplete in the cells, maybe the phosphorylation comes from unlabeled BTK, we did this experiment where after labeling with 4 of our compounds, we added ibrutinib. And you see that when we add ibrutinib, without these probes, we completely eliminate BTK phosphorylation, but with our probes, we're able to protect BTK from ibrutinib and preserves its activity in cells, showing both that our compounds label the cysteine that's required for ibrutinib to bind through, and that they keep the protein active. We also showed this through the primary B-cell assay, in which 2 of our compounds are completely inactive, despite, as I showed you, being able to label in these cells, whereas ibrutinib inhibits the B-cell activation. To try and take advantage of the fact that the active site is free for other compounds to bind, we wanted to see whether we can use our tags as sensors for targeted degradation. So in this experiment, we took a probe, we installed, using colder chemistry, a fluorophore on BTK, leaving the active site empty, and then we used a PROTAC for BTK, a reversible PROTAC, to recruit an E3 ligase that would ubiquinate BTK, degrade it, and we can follow this process by the fluorescence of the tag. So we used this PROTAC, 1Q, a reversible BTK PROTAC, and this colder BODIP probe. And you can see here that adding only the fluorophore, we can see the fluorescence band for BTK. Adding the fluorophore and the PROTAC, we get about 50% degradation by fluorescence, and also by Western blot, we get about 50% degradation. So this is a good reporter for the amount of degradation. And then if you only add the product, of course, you don't see anything in the fluorescence channel, but you get the same % of degradation as with the fluorophore, meaning that the tag did not interfere with the degradation and is a good reporter for that process. And here you see the quantification, which basically says the same thing. We also wanted to see whether we can directly degrade BTK using this chemistry. So now, instead of putting a fluorophore as the tagging group, we put a E3 recruiting element, a thalidomide analog, as the tagging probe, and then this would label BTK irreversibly, recruit E3 ligase for ubiquitination, and degrade the protein. We made a small series of different linker lengths and different analogs of thalidomide. You can see by LCMS that they all label BTK very rapidly. Within 10 minutes, we get close to 100% labeling. And here you can see the degradation results. So basically a very potent degrader with a DC50, the concentration at which 50% of BTK is degraded is much lower than 100 nanomolar. We did some controls showing that ibrutinib can block the degradation. Also, thalidomide can block the binding to the E3 ligase and block this degradation. And using chemoproteomics, we showed that BTK is the most prominent degradation target, gets lowered by more than 16-fold, with only 2 off targets that are significantly degraded. CSK is an off target of ibrutinib, but is only degraded by about 50%. And ER3A is also known as GSPT1, which is an off target of the cereblon binding molecule, so not of our compound. So a very potent and selective degrader. So in summary, I showed you that using colder chemistry, we can site-specifically label the protein, a specific cysteine in the protein. There are other reported ligand-directed chemistry for tagging, but they typically are not specific to a single type of amino acid, and they're more random. And here we can select the site of modification. I didn't show you, but we verified that by LC-MS trypsin digestion experiments. These are tunable, and we can attenuate their selectivity. They preserve the protein activity, and as such, we think they're a very exciting platform for new types of proximity inducers. So I already showed you PROTACs, but you can easily think about phosphorylation-inducing chimeras. Since we keep BTK active, we can now recruit neo-substrates to BTK and turn it into a new phosphorylation-inducing enzyme. With that, I want to acknowledge my entire lab, and specifically Ram, who was the chemist responsible for everything you saw in this presentation. Efrat and Nadi were responsible for the biological validation and cellular work, and Ronen was responsible for all the chemoproteomics and proteomics experiments. I want to thank you for your attention, and I'm happy to answer any question. And since I know this is specifically a meeting for young chemists, I want to invite anyone who's interested in this type of chemistry. We have postdoc positions available. The Weizmann Institute is very international, and we'll be happy to accept application from people who are interested in covalent chemistry. Thank you very much, and I'll stop my sharing now.
So thank you, Dr. London, for your brilliant results and presentation regarding this new covalent actors, and for your also for thinking on the future about the covalent inhibition of kinases. So let me check if there are some questions from the audience. Oh, maybe one. So there's one from Dr. Francesco Di Leva. That is one of our organizers from the University of Naples. So so Francesco, thanks for your very kind, very bright presentation. Very much for the good presentation. He was wondering if you also investigated the efficacy of your ibrutinib analogs against the BTK mutant strains, which are resistant to the drug, to the original drug.
So we haven't, but I'm pretty sure that any mutation to the cysteine, so one of the first mutation that arises against ibrutinib is a cysteine 481 serine mutation. And I suspect any such mutation would render our compounds inactive, or at least reduced by about 100-fold their potency. So this very heavily relies on the cysteine. So if you think about it from a therapeutic standpoint, these compounds might be more suitable for non-cancer indications, such as rheumatoid arthritis or any sort of inflammation related disorder where BTK is a target, and not for cancer where resistance mutations arise very often.
Okay, so there are no further questions, but I have no, maybe there's another one. No, sorry, it's the same. Sorry, I have some remarks or comments and some curiosities. The first one is, okay, so if you have any ideas of the two opposing mechanism of these metacrylates analogs? I mean, this can be can undergo addition or substitution according to the living group. And of course, the reactivity is directly related with the pKa or the pKb of the living group. But my question is, if you have ideas or results regarding why some living groups act in a similar way, so giving you just a substitution, and others just giving you the addition. And if you consider this when you design the dual BTK inhibitors attached with the metacrylamides.
Yeah, so very good question. So far, we don't have enough data to understand perfectly why some of the compounds act through substitution, some through addition. We do see a trend where amine-based compounds tend to undergo this mix, so 50% substitution, 50% addition, whereas oxygen-based compounds are almost always substitution. So that's one rule of thumb. We do know it also depends not only on the heterosubstituent, but on the properties of the amine on which the acrylamide is based. So for instance, in ibrutinib, the amine is coming off of a piperazine moiety, whereas in our model compound, it came off of a benzylamine. So modifying that amine also influences the leaving ability. So in the reverse code, in the second half of the talk, all of the compounds were based off of ibrutinib on this piperazine, and then we always saw leaving of the piperazine. So whereas in the other direction, it was 50-50. So I think there's a lot to learn, and we're accumulating slowly examples of these types of chemistry. And I think we'll be able in the future to derive design rules for when which reaction will happen and what will be the reactivity.
Okay. So another very last question is if you think that it's possible to use these metacrylates to deliver type 1 and type 2 kinase inhibitors, because basically you use ibrutinib, and I was thinking if you can use this not only for kinase inhibitors but also for other targets in order to have a dual inhibitor and that can switch off the proliferation of a cancer cell if this proliferation is linked to these two targets.
Yeah.
Both.
So I mentioned it very briefly in the summary. We are working on, for instance, using, for instance, KRAS G12C is only expressed in cancer cells, right? And cysteine is not found in normal tissue. So using molecules targeting this cysteine to release a toxin, for instance, that would only get released in the cancer. Or you can think of other inhibitors that synergize with KRAS inhibition, such as EGFR inhibitors or AKT inhibitors. So releasing a second inhibitor upon binding and inhibiting your target protein. So this is very early days, but we are thinking in that direction. So it doesn't have to be kinase inhibitor. Whenever you have so for instance, we already showed that it worked for PL-PRO, which is a protease. So wherever you have a suitable cysteine and a scaffold that can direct to it, we think we can utilize this strategy.
Okay, thank you. Thank you. So I would like to thank you once again for your presentation, for being with us today. And I really congratulate with you for your very brilliant results and research. Thank you very much.
Thank you.
So now I think it's the time. We are just 5 minutes in delay. I think it's the time to start with the oral communications. And now we will proceed with Dr. Giulio Poli from the University of Pisa, that I see already on the screen. Dr. Poli will present his presentation, sorry for the word, called Novel Cell Active PIN1 Inhibitor Identified Through Pharmacophore-Driven Virtual Screening. So Dr. Poli, welcome, good morning, and the stage is yours. You can start whenever you want to.
Good morning to everybody. I'm Dr. Poli from the virtual screening, molecular modeling and virtual screening laboratory of the University of Pisa. I hope you can see now the presentation and hear me properly. Can you confirm?
Yes, you see everything and we hear you clear.
Okay, so yeah, wait a second. Yeah. Okay, so first of all, I want to thank the scientific committee for selecting my talk, which deals with the development of novel PIN1 inhibitors with cellular activity through a pharmacophore-driven virtual screening study. So PIN1 enzyme catalyzes the cis-trans isomerization of peptide bonds and terminal to proline residues next to either phosphocserine or phospho-3 residues, and thus regulating folding and maturation and proliferation of different kinds of cells. So PIN1 is involved in many different cellular and pathological processes, upregulating more than 50 oncogenes and inhibiting more than 20 tumor suppressors. And for this reason, PIN1 overexpression has been observed in many different types of cancer, and especially it was recently found overexpressed in high-grade serous ovarian cancer, for which it is considered a promising target. And most of the available PIN1 inhibitors available to date suffers from poor cell permeability that is due to the presence of phosphate groups or multiple negatively charged groups in order to mimic the substrate negative charge. So the identification of novel PIN1 inhibitors is still a hot topic in medicinal chemistry. And thus in this context, we developed a receptor-based virtual screening starting from the X-ray structure of PIN1 in complex with the phenylimidazole inhibitors that was used as a starting point for the development of cell-active inhibitors. So by inspecting the X-ray co-crystal structures, we identified the key binding features of the fragment ligand to the key anchoring residues of the enzyme. And in this way, we developed a receptor-based pharmacophore model taking into account all these interactions. There is 4 different hydrogen bond acceptor, 1 H bond donor, and an aromatic residues. This without using a negatively charged features in order to foster the identification of compounds with only 1 negatively charged at maximum. So this pharmacophore model was used to screen a large database of more than 5 million commercial compounds. And only compounds matching all pharmacophore features, that is the 5 H bonds and the aromatic features, were eventually retained and considered for further studies. So about 5,600 compounds were subjected to a docking strategy, and especially the consensus docking evaluation. So let me spend a few words about the consensus docking approach, which consists in using multiple docking methods and at the same time in combination in order to improve docking reliability in both a quantitative and a qualitative point of view. So the rationale behind the strategy is very simple, and that is, if the same ligand binding pose is predicted by multiple docking methods, this binding pose should be more reliable than other binding poses predicted by only single docking methods, as in classic docking studies. So how this approach works? Basically, every single ligand that is subjected to the consensus docking approach is docked within the studied target by using multiple different docking procedures. For example, 10 different docking procedures or even less. And then the reciprocal distances in terms of root mean square deviation between all different docking poses produced by for each ligand are calculated. And after setting an RMSD cutoff, normally of 2 angstrom, the different binding poses are clustered together. And so the most populated cluster of poses with reciprocal distances below the cutoff is identified. And in this way, it is defined and identified the so-called consensus docking level. That is the number of docking poses within the most populated cluster of poses, which also corresponds to the number of docking procedures producing the same binding mode. And here we can see a couple of examples. The example of a consensus level 5, in which 5 out of the 10 docking procedures used produce the same binding mode, and an example of a maximum consensus level, in which all different docking procedures produce the same binding mode. So by using cross-docking studies, we demonstrated that the higher the consensus level obtained for a certain ligand, for a certain binding mode, the higher the reliability of the predicted binding mode with respect to experimental binding dispositions. Also, by using enriched databases, multiple enriched databases, we demonstrated the ability of consensus docking and discriminating active ligands from decoy compounds, of inactive compounds. In fact, the higher we demonstrated that compounds for which a high consensus level is obtained when docked within the corresponding target are more likely to be active compounds with respect to ligands for which a low consensus docking is obtained. And for this reason, consensus docking has been applied in combination with other techniques within multiple virtual screening strategies that were that successfully helped us in identifying different hit compounds for different targets. For example, fatty acid amine hydrolase, the first star D3 inhibitor ever reported, and one of the most inhibitors of Mycobacterium tuberculosis salicylate synthase. A particularly efficient implementation of consensus docking that was used in the last strategies is what I call pharmacophore-driven consensus docking, in which a receptor-based pharmacophore model is used for pre-filtering a large commercial dataset of compounds. Then the pre-filtered compounds are subjected to the consensus docking analysis, and then the compounds for which a high consensus level is obtained are passed again through the pharmacophore model in order to identify compounds with reliable binding modes that still match the pharmacophore requirements. In this specific case, in this specific virtual screening study, we used a hierarchical pharmacophore-driven approach in order to further speed up the calculation. So we first apply the PIN1 receptor-based pharmacophore model to pre-filter compounds. The pre-filter compounds were docked using the fast docking methods among 5 reliable docking methods for docking PIN1. Then the compounds were post-docking filtered using the pharmacophore model. The retained ligands were then docked using the second docking method and then passed again to the post-docking filter, and so on, in a systematic approach and a hierarchical approach by using 5 different docking methods. At the end, the compounds for which all 5 docking poses respected the pharmacophore features were then subjected to the consensus docking analysis in order to identify the final set of compounds. So by using this procedure, out of the 5,600 compounds, only 10 compounds for which a minimum consensus level of 4 out of 5 was obtained, and for which all docking poses respect the pharmacophore filter, were retained and further evaluated using molecular dynamic simulation for assessing the stability of the binding modes. And only 2 compounds for which a ligand root mean square deviation during the MD below 2 angstrom was obtained, and maintaining at least 3 out of the 5 H bonds represented by the pharmacophore model for at least 70% of the whole simulation were eventually retained and subjected to biological evaluations. PIN1 inhibition assays demonstrated that both identified compounds showed PIN1 inhibitory activity in the micromolar range and with comparable or even higher potency with respect to all trans-retinoic acid that was used as a reference compound, since it emerged as a cell-active PIN1 inhibitor. Unfortunately, anti-proliferative assays over 3 different ovarian cancer cell lines show that the most active compound VS1 was not cell-active, but however, compound VS2 demonstrated a very promising cellular activity, either comparable or even 2-fold or 6-fold higher than all trans-retinoic acid, in particular over against ASCOV3 cells. And we hypothesized that this different cellular activity of the compound was due to the different lipophilicity of the ligands. In fact, for compound VS2, a higher partition coefficient at pH 7.4 was predicted with respect to compound VS1. So here I show you the predicted binding mode of compound VS2, and as you can see, the ligand properly respects all the H bonds with the key anchoring residues of the enzyme, and also presents an additional H bond with the histidine 59. And being a small molecule with low molecular weight and low heavy atoms, it represents a very promising starting point for the development of more potent and cell-active PIN1 inhibitors. So in conclusion, we developed an efficient virtual screening strategy, combining receptor-based pharmacophore modeling and hierarchical consensus docking. We identified 2 new PIN1 inhibitors that validated the reliability of our in silico strategy, and a novel cell-active compound that represents a valuable starting point for hit to lead and lead optimization studies. So we're actually going to develop derivatives of compound VS2 for identifying more potent analogs, and we're going to apply this hierarchical pharmacophore driven procedure to other and even larger commercial database in order to expand the chemical space of PIN1 inhibitors. So before finishing, I want to thank Professor Tiziano Tuccinardi from the University of Pisa, but also Professors Minuto, Logranchi, and Macchia, which contributed to this work, and also special thanks to Dr. Flavio Rizzolio from the University of Venezia, and also Professor Canzonieri and Dr. Caligiuri that performed the experimental part. So, and of course, I want to thank you for your kind attention. Thank you so much.
Thank you, Dr. Poli, for your presentation and for being with us today, and also for being right on time. I think we have we have a question directly from near London. Dr. London, congratulates with you for your methodology. And he says, I noticed that the final VS2 still contains the carboxylic acid. I thought the major design consideration was to remove the negative charge, wasn't it? So.
Okay, I thank Dr. London for the question. Yeah, actually, the major design consideration was to identify compounds not endowed with phosphate groups and with a single negative charge, because actually the reference inhibitors used co-crystallized with PIN1 with 2 carboxylic acid was successfully used as a starting point for cell active compounds. But actually the reference compound itself was not cell active due to the presence of 2 different carboxylic groups. So there are actually compounds endowed with a single negative charge that are cell active. But of course, compounds with phosphate groups of double negative charge are not cell active. So we wanted to try to identify compounds with either a negative charge or without negative charge endowed with cellular activity. So as shown by the biological evaluation of the 2 hits, we actually identified 2 inhibitors with promising enzyme activity, but only one of which is cell active. I mean, both include a negative charge, but with a different cellular activity. So it is possible to obtain cellular activity with only one single negative charge. Of course, if the structure of the ligand allows sufficient lipophilicity or sufficient cellular permeability. So we wanted to try this approach. Of course, we will aim at developing more active derivatives of compound VS2, increasing the lipophilic portion of the ligand. So by reducing the total polar surface area in order to improve lipophilicity and also cellular permeability. This is definitely.
Okay, thank you. We hope that your answer will satisfy Dr. London. We have other 2 questions, one from Dr. Coluccia, one from Dr. Brindisi, both from the scientific committee. I will start with Margherita, if Antonio agrees with my first.
Yeah, it's fine, of course.
Okay, so Margherita, okay, very nice talk. She said, I have a question. Did you evaluate experimentally or computationally the selectivity over PIN4?
So I thank Margherita Brindisi for the question. Actually, no, we did not evaluate the selectivity over PIN4 isoform, but that's that's a good point. So we may evaluate the selectivity either, first probably computationally and later on experimentally over this isoform. That's a good point. Thank you for this question.
So Antonio, if you want to add some other questions.
Hey, thank you. Hi, Giulio, very nice presentation. Just a curiosity, you showed that you use more than one software for the docking evaluation. There is an established order, so you may use always before Autodoc and then Gold and then Vine and so on.
Yeah, thank you for the question. That's really interesting. Actually, in this case, so in the case of a hierarchical consensus docking approach, there is actually an order that is based on the speed of the different software. So for maximizing the efficiency of the hierarchical procedure, we have to use first the fastest procedure, then the second procedure in order of speed, and then the slowest one at the end, so that a smaller and smaller number of compounds are docked with the slower and slower procedures. So this is to maximize the efficiency of this approach.
All right. Thank you. Just a curiosity, it sounds really time consuming. Which machine did you use?
We use Intel computers with up to 6 processors.
All right.
But actually we use different machines and different processors.
Okay.
Of course, the most time consuming part is the consensus docking. That's why we wanted to try, we applied consensus docking many different times, and also the pharmacophore-driven consensus docking, but we wanted to further speed up the procedure by using a hierarchical approach. Of course, if you use a hierarchical approach, it's better to use a lower number of docking procedures, because you might end up with no molecule at the end, passing all different filters.
Yeah.
So you reduce the number of procedures and reduce the number of compounds docked with every procedure. So, the advantage in terms of computation time is very impressive.
Thank you very much.
Thank you too.
So once again, thank you, Dr. Poli, for your presentation and for your contribution. So
thank you very much.
And there are any other questions, you can contact him in private on our chat. So we need to move on, and I think it's the time for Dr. Simona Mezzella from the University of Salerno in Italy. She will present her talk entitled Design and Synthesis and Biological Evaluation of a Dual-Acting SARS-CoV-2 Protease Inhibitor Agent. So Dr. Mezzella, good morning and welcome. The stage is yours, you can start.
Okay.
We see your screen.
Okay. Good morning, and thank you for your kind presentation and for giving the opportunity to discuss this work, focused on pharmacological workflow, allowing the identification of a dual-acting SARS-CoV-2 protease inhibitor and a selective spike protein inhibitor. SARS-CoV-2 is the pathogen caused the coronavirus disease in 2019, and since this date, it has caused the worldwide outbreak. Its genome is a positive sense RNA strand codifying for structural and non-structural proteins, of which 4 play a crucial role in the infection cycle. These proteins are the well-known spike protein, the RDRP, responsible for the viral replication, and the 2 different proteases called main protease and papain-like protease involved in the protein maturation process. So the pivotal role of these enzymes make them the most investigated, the druggable pharmacological target to contrast SARS-CoV-2, in particular main protease. Two different classes of SARS-CoV-2 and Pro inhibitors have been identified so far. Covalent inhibitors, including different molecules that share some four features consisting in a moderate sites, a group mimicking glutamine side chain, a lipophilic group, and a reactive electrophilic warhead responsible for the covalent interaction in the binding site. Non-covalent inhibitors, including different flavonoids such as bicalane. So from this evidence, we were aimed to identify a new hit compound, both covalent or non-covalent SARS-CoV-2 and Pro inhibitors using a rational design approach based on molecular modeling calculations. In 2020, it was reported the structure-based identification of two different SARS-CoV-2 and Pro inhibitors, A1 and B1, with can react with cysteine 145K residue due to the presence of aldehydes moiety responsible for the covalent interaction. As shown in this picture, cysteine 145K residue is placed in S1 first site, while S1 and S2 can accommodate different large and cyclic systems, while S4 site was prone to interact with indole moiety to form hydrogen bonds. So, starting from these evidence, we present the design, the synthesis, and the pharmacological characterization of peptidomimetics. Using L-tryptophan, we introduced the different amines and natural modified amino acids to obtain pseudopeptides. Two other molecules were synthesized using methylindole 5-carboxylate, derivatized in positions 1 and 3 to obtain the corresponding 3-aminomethyl-1 substituted derivatives. Our workflow is based on molecular modeling calculations, leading the synthesis of different compounds, driven step by step by the outcomes obtained from enzymatic assay. So recursive cycles of in silico design, synthesis, and biophysical analysis of target engagement were performed. And in this way, the combination of in silico and experimental testing led the gradual generation of a small library of compounds that share some similar chemical functions, consisting in a lipophilic substituent with the different moderate, different steric sites, an electrophilic moiety potentially responsible for the covalent interaction with cysteine 145, and an amino acid backbone to form hydrogen bonds. The synthesis of these compounds is here summarized. A coupling reaction with the L-Boc tryptophan with different amines, followed by a Boc deprotection and a coupling reaction with different Boc amino acid, gave the final derivative 7 and different pseudopeptide intermediates. Then a new Boc deprotection followed by reductive amination with different aldehydes gave the final derivatives. Similarly, using two different aldehydes and starting from pseudopeptide intermediates, we obtained final derivatives 25 and 28, and on the other hand, an acylation reaction with the chloracetyl chloride gave the final compound 29. Then, using a methylindole carboxylate, an alkylation reaction firstly was performed, then a Mannich reaction followed by an alkylation reaction with benzyl bromide or a coupling reaction with the propylglycine with final derivatives 34 and 35. So in order to confirm these are clues obtained by molecular modeling experiments, using MPRO, enzymatic assays were performed, and as shown, compounds 7, 23, 27, and 29 were the most potent compounds. And this is in accordance with the molecular modeling calculations that showed non-covalent poses for compounds 7, 23, and 27, while compound 29 seems to bind covalently MPRO. So in order to expand the obtained enzymatic results, the synthesized compounds were tested using verocells transfected with 2 different clinical isolates SARS-CoV-2, and despite the remarkable antiviral activity observed in enzymatic assays, compounds 7 and 23 failed to inhibit the viral plaque formation, while compounds 27 and 29 confirmed the antiviral cellular activity, and in particular compound 29 antiviral potency. It was compared to remdesivir, and it was 28-fold higher than compound 27. While compound 34 that was unable to inhibit MPRO in enzymatic assays showed an interesting antiviral cellular activity. So we questioned if the synthesized compounds were able to inhibit also other SARS-CoV-2 proteins, and for this reason, PL-Profret and spike protein SPR assays were performed. And we observed that compound 29 showed remarkable affinity also against the PL-Pro. So its dual inhibitory effect is likely responsible for the antiviral cellular activity observed in cellular assays. And interestingly, we found that the antiviral activity, cellular antiviral activity observed for compound 34 is due to its ability to inhibit selectively spike protein. In order to rationalize this data, molecular docking experiments for compound 29 against the PL-Pro were performed, and 2 different models related to non-covalent and covalent binding mode were generated. So the output poses arising from a covalent binding model highlighted a different binding mode, and in particular, indole and benzyl functions were oriented toward the external part of the protein. Then also molecular docking calculations between 34 and spike protein were performed. The accommodation of 34 highlighted a good shape complementarity with the external part of the protein, and we observed then some K-interactions, a network of interactions with different residue responsible for the spike AC2 protein-protein interactions were retained. In conclusion, the simultaneous proteases inhibition can significantly hamper the SARS-CoV-2 machinery, the viral machinery, and represent an interesting therapeutic option to SARS-CoV-2 tackling, making compound 29 an interesting hit compound for the development of a new class of anti-SARS-CoV-2 agents. On the other hand, the extended investigation led to the identification of compound 34 as a potential selective spike protein inhibitor. I'd like to thank the scientific committee, my colleagues, and all you for your attention.
Thank you, Dr. Simona Mosella, for your presentation and for your for being in time. We have. Let me check if we have some questions in our chat. Yes, we have. So there is a question by Dr. Sveva Pelliccia. She said, "Thank you for your nice presentation. I was wondering about the possibility to change your warhead with other. Have you explored this aspect?"
Yes, we did. We changed it with the different warhead, and the best group, it was the chloracetyl group to react covalently with the cysteine 145. We used alkane, alkene, and other group.
Okay, so thank you for your answer. We hope that this answer will satisfy Sveva. And so I think we are, I think it's the time for the break now. And so we will have 10 minutes. Thank you once again for Dr. Simona Mosella from the University of Salerno. And so thank you to all the attendants for their for being with us. So we will have 10 minutes, more or less 10 minutes of break, so in order to recover, to spare some time. We were just 5 minutes in delay, and so we will see around about 11:30 more or less. Okay, so thank you once again to all the speakers, from the plenary lecture to the invited speaker and all the other oral participants, and to all the attendants for their attention. So thank you. See you later. So, do you hear me? Someone? Yes, I see already Dr. Antonella Messore. So welcome back to the AMYC-BIOMED 2021. So we have came back from the coffee break, and so now it's the turn of our third speaker of today. That is Dr. Simona Mosella from, Dr. Antonella Messore from Sapienza University of Rome. Her presentation is entitled Discovery of Non-DKA Derivatives Inhibitors of the HAV1 ribonuclease H function of reverse transcriptase. So, Dr. Messore, welcome. And so the stage is yours. You can start. Good morning.
Sorry, Antonella, you have to turn on the microphone.
Yes. Okay, now
you can.
Yeah, we also hear you.
Ah, okay. Because I didn't change nothing, though I don't know why you cannot hear me before. Okay.
So you can start.
Okay. The human immunodeficient virus, HAV1, is the causative agent of active immunodeficient syndrome, AIDS. The viral core of the virus contains the genome of HAV1, composed of 2 identical copies of single-strand RNA that contain 9 genes. The 4 genes encoded for 3 proteins: integrase, reverse transcriptase, and protease. In total, 47 FDA-approved medicines can be used in the treatment of HIV. This approach is called antiretroviral therapy. Despite the relevance of this approach still has several drawbacks, including both acute and chronic toxicity and the inability to eradicate the virus that causes lifelong treatment and drug-resistant form. Since the discovery of HIV-1, reverse transcriptase has been the first explored viral target due to its critical role in viral replication. This enzyme shows 2 domains, polymerase domain and endonuclease domain, referred to as RNase function. The active side of this function shows a very high conserved DEDD motif, able to chelate 2 magnesium cofactors that are essential for the activity of this enzyme. A similar arrangement is also present in another metalloenzyme, integrase, that play a critical role in viral infection. Currently, all approved RT inhibitors are targeting the polymerase function of this enzyme. In the field of antiviral agents, the decatoase derivatives are the first reported chelator of magnesium cofactor able to inhibit integrase, like BDDDA that you can see here. Thanks to the high similarity between the active side of RNase function and integrase, several of these compounds were also tested against RNase function and found to be active. In this field, my research group has previously reported several decatoase derivatives as a dual inhibitor of integrase and RNase function, and the integrase inhibitor endowed with marginal activity against RNase function, like you can see here in this slide. Despite the relevance of decatoase chain inhibitory activity, it is also well known that this posture suffers several pharmacokinetic limits, such as high metabolic turnover and poor cell membrane penetration. For this reason, several compounds were developed by mimicking this portion into a couple with better pharmacokinetic properties. In fact, about that, a success case in the HIV field was the discovery of raltegravir and elvitegravir, 2 integrase inhibitors. Keep in mind this kind of approach, so we design new series of compounds with the aim to overcome the limits of this portion. In detail, we design to retain the chelating portion, and regarding the quinolinonyl derivative, the decatoase chain was short in carboxylic acid function that, together with the ketone group in pole position of the quinoline ring, are able to chelate the magnesium cofactor. In the field of pyrolyl derivative, we decide to mimic the alpha-gamma decato portion into a parallel ring. Let me go in deep regarding the new design and then synthesize compound. For the quinolinonyl derivative, we decide to fix the parafluorobenzyl group in 1 position and change the substituent in 6 position. with different modes of substituent, but also with different ether group, characterized by different degree of freedom and steric hindrance. Here, I just report a general synthetic route for the phenol-enol derivative. We start from the condensation of compound 7 with the ethyl-ethoxymethylammalonate, the compound 8, that was submitted to thermical reinclusion to give compound 9K. Same condition was also used for the synthesis of all 9 intermediates, starting from the commensurate amine. The ester derivative 6 were synthesized by acylation in presence of parafluorobenzylbromide in alkali medium for potassium carbonate. And in the end, a base catalyzed hydrolysis supported the corresponding acid derivative. All tested compounds are all synthesized compounds are tested in vitro to evaluate the ability as a NITRAC inhibitor, and you can see here the results. The best compound of the serial compound 5 and 6O that show a comparable EC50. The acid derivative were generally more active than the corresponding ester counterpart, and in general we can say that an increase in the dimension of substituent 6 position affords compound with high activity. To evaluate the antiviral activity, the best compound were also chosen to be tested on cell-based assays, and I report in this table the results. The acid derivative show the best activity, with the compound 5C that showed the lowest EC50. Thanks to the RH inhibitors are also able to inhibit other viral targets, we decided to test the best compound also against the polymerase function of RT and integrase. Like you can see in this table, 7 compounds were found selective for their RT function with respect to integrase, and 2 compounds were found very active against integrase. We didn't not observe the selectivity for the polymerase function. Only 2 compounds show a very low selectivity for RNase function, and one of that was also one of the best compounds of the series. In the light of this important result, this compound, together with the compound 5T that showed the lowest, the best antiviral activity, were chosen for the docking study. In our model, the oxygen atom of ketone group, together with the carboxylic function, are able to chelate the metal cofactor in the active side. Moreover, both compounds are able to stabilize different interactions with very high conserved amino acid residues. To experimentally verify the binding modes tested, by the docking study, the best compounds were also tested against several points of mutants of HIV1. And like you can see here in this slide, the mutageneity experiment confirmed the binding mode suggested by the docking study. In the end, we also evaluated the importance of coordination between the active compound and magnesium cofactor, so a spectrophotometric complexation analysis was performed. A treatment of compound 6O in presence of magnesium chloride resulted in an increase in the absorbance. So this means a coordination between the active compound and magnesium cofactor. We were also able to calculate the stoichiometric ratio that was 1:1. Similar result was also obtained for the acid derivative 5O. About the second scaffold, also here we decided to keep fixed the pharmacophoric portion and change the substituent 1 for position. So we obtained 2 series of compounds. The first with different substituent 1 for position, the second with different oxyphenyl group characterized by different degree of freedom steric hindrance. The generic synthetic route for the parazole derivative is applying the scheme 2. The synthesis of N substituent pyrolyl derivative 17, starting from the corresponding pyrolyl derivative with the appropriate alkyl halide in alkali medium for potassium carbonate. Then applied the condensation for the corresponding ketoester derivative 18. Same condition was also used for the other starting material that you can see here in the slide. And then a chlorpyrazole synthesis in presence of hydrogen hydrochloride in acid condition for the corresponding ester derivative 13. In the end, a base catalyzed hydrolysis for the corresponding acid derivative. Also in this case, all synthesized compounds were tested in vitro to evaluate the activity as RH inhibitor, and you can see in this table the result. The compound of the series 14 and 15 showed the best activity with the IC50 in low micromolar, submicromolar range. The best compound is compound 40B that show an IC50 of 0.27 micromolar. The best compound were also chosen to be tested on cell-based assay and are reporting stable the results. In this case, the ester derivative showed the best activity with the compound 38 that showed the lowest EC50. Like here, the best compound were tested against the integrase structure related with the RNA function, and like you can see, all tested compounds are selective for RNA function respect to integrase. To clarify, the biological tests of the study were performed. In our model, the nitrogen atom parasol ring together with the carboxylic function are able to chelate the metal cofactor in the active side, and all compounds are able to stabilize also here a lot different interaction with very high conserved amino acid residues. As upper for the quinolinonyl derivative, mutagenesis experiment confirm the binding mode suggested by the docking study. In the end, we also evaluate if to investigate also if our strategy of replace the decay chain with a parazole ring results in more stable compound. So liquid chromatography and spectroscopic analysis were performed. In particular, we checked the stability for the compound 6010, a decay ester derivative, and its corresponding parazole analog 30A and the corresponding acid counterpart 12A in 90% of human serum at 37 degrees. Like you can see here in this picture, the compound 6010 starts to be unstable after 3 hours. The corresponding ester counterpart was found completely unstable. We were also able to identify the degradation process that could be ascribed to retroglycan adduct. On the contrary, like you can see, the new synthesized compound 30A and 12A were perfectly stable for all duration of the experiment. So this means that our strategy was successful. In conclusion, a new anti-HIV agent are still needed. Here I show you a new series of quinolinonyl and pyrroline non-decayed derivatives as a result of isosteric approach of previously decayed derivatives. The docking study highlights the ability of this compound to interact with the magnesium cofactor in the active side, and also the ability to stabilize different interactions with very high conserved amino acid residues. Moreover, the new synthesized compound should also promising serostability with respect to the corresponding decayed acid derivatives. It's the time to say thanks. First, I want to thank all my research group, in particular Professor Roberto Di Santo, Professor Roberta Cosi, and Professor Luigi Scipione, Professor Enzo Tramontano and Professor Medifio for the biological results, and Professor Sandro Tosconati for the docking study. And obviously all of you for your kind attention.
Thank you, Antonella, for your presentation.
Thank you.
And I'm gonna check if there are some questions from the audience. Yes, there's one from Francesco Di Leva. He said, hi Antonella, thank you for your nice presentation. Since the pyrrole ring of your derivatives might be involved in metal chelation, I was wondering if you investigated its pKa experimentally or at least computationally.
The in
pKa.
Okay. Yes, so all calculation was, so I didn't, the calculation of the docking was performed at pH 7.4. So this means that probably the carboxylic acid, so the carboxyl, so is negative charge for the carboxylic function in the case of the quinolinonyl derivative. For the pyrrole, it's the same. The pH was 7.4 for the calculation. And
okay, so thank you once again for your presentation, Antonella, and now it's the turn for the next speaker. So I would like to welcome to Dr. Piermichele Cubari, that is already online. I see you on the screen. Hi. So Piermichele Cubari is coming from the University of Groningen in the Netherlands, and it will present a talk about the rational design of light-controlled bioactive compounds for photopharmacology. So Piermichele, welcome, and feel free to start. We see already your screen. So
perfect. Okay, can you hear me?
Yes.
Yeah. Okay. Hi, everyone. I'm Michele Cobauri. Thank you for the kind introduction. I'm a PhD candidate at the University of Groningen, and also thank you for the opportunity to present my work here. I would talk about rational drug design for photopharmacology in my presentation. But first of all, what is photopharmacology? Well, photopharmacology is a novel approach in medicinal chemistry that uses light to control the bioactivity of drugs. This control is obtained with photoresponsive units such as molecular photoswitches. And the most commonly used photoswitch in photopharmacology is azobenzene, which isomerizes from the stable trans isomer to the metastable cis isomer upon irradiation. The ideal scenario for any application in photopharmacology requires an inactive trans form of the drug, which can be activated with light only where and when needed. And this would result in low adverse effects and low environmental toxicity for the trans isomer, which then we can activate with high spatiotemporal precision and regulating this, the dosage with light. Then the drug will relax thermally, or we can switch it back to the inactive trans isomer with another wavelength. But photopharmacology is interesting and a very cool approach because can potentially increase site selectivity in medical applications. So, for example, here I'm showing you in one of our pioneering studies, our group developed photoswitchable antibiotics, which can help in the fight against antibiotic resistance. This approach would reduce the interference with good bacteria in the body and also reduce the buildup of active antibiotic in the environment. This molecular design I'm showing you here was based on the class of quinolones, and the introduction of an azobenzene into the drug scaffold enabled photocontrol of antibacterial activity with extremely high precision, as you can see from the Petri dish on the right. This serves as an example of one strategy for rational design in photopharmacology, which is called Azo Extension. In this approach, we consider structure-activity relationships of the original drug, and then we attach the azobenzene where different substituents are allowed or tolerated. So in this way, we hope to achieve photocontrol of drug potency with the addition of this photoswitch. Another strategy for rational design in photopharmacology is called Azologization. In this approach, we introduce the azobenzene in place of an aromatic moiety that is sterically and electronically similar. And these moieties, which are normally two aromatic rings linked by a two-atom linker, are called azosters because of as a mixture of azobenzene and biazoster, of course. So, as we already discussed, in most applications, photopharmacology aimed to design photoswitchable drugs that are more active in the metastable state. So we call them cis-on in the case of azobenzene. However, most attempts at azologization normally result in trans-on ligands, especially when we substitute planar azosters that are and adopt a transoid conformation. So an explicit set of general criteria for the design of cis-on photoswitchable drugs is still missing. And here we present the identification of the biaryl sulfonamide scaffold as a cisoid azoster because of its structural and electronic similarity with cis-azobenzene. So first, we analyzed the Cambridge structural database, and this provided insights into the requirements for a cisoid geometry. We see that the biaryl sulfonamide have a similar centroid angle, as you can see from the distributions of the centroid angles on the left, and the overlap between the green distribution and the red distribution. But also a ring distance is much closer to the one of cis-azobenzene than the one of trans-azobenzene. Also, we explored the electronic similarity with DFT calculations, and we see that if we compare it with trans-azobenzene, Cis azobenzene approximates better the dipole moment of bioyl sulfonamides and their electrostatic potential surface. So we then hypothesized that the azologization of bioyl sulfonamides would be a general strategy for designing ligands that are more active in the cis state. However, of course, we are replacing, by doing that, a moiety that has 2 hydrogen bond acceptors and 1 donor in the linker. And we substitute this with our azo bond that only has 2 acceptors of hydrogen bonds. So we have risks of loss in affinity with the target. So yeah, to test our hypothesis and its limitations, we took 2 bioactive compounds as 2 examples and investigated them by molecular modeling, synthesized them, photochemically characterized them, and also we evaluated their bioactivity. So the first example is a nanomolar inhibitor of lipoprotein-associated phospholipase A2, which is a potential target for atherosclerosis. So we first studied the hypothesis in silico. And on the left, you can see that induced fit docking suggested that the cis isomer would mimic much better the binding mode of the original ligand in green, and especially if compared with the trans isomer that is docked, and it's the blue structure. So on the other hand, the cis isomer, it was not able to form the hydrogen bonds that are important for the affinity with the oxy anion hole that is represented there. We also performed 100 nanoseconds of molecular dynamic simulations that showed that the binding mode of cis-azo1 in red was also much more stable over time when compared to trans-azo1. So the photochemical properties of azo1 were investigated in buffer, and we irradiated with light of 365 nanometer, and this resulted in the photoisomerization from trans to cis, while for 20 nanometer light promoted the back isomerization from cis to trans. The compound showed no fatigue upon multiple cycles of photoswitching under reducing conditions. And moreover, we saw that the irradiation with 365 nanometer light promoted the enrichment of our cis population to a very high photostationary state distribution, PSD, of 92%. And finally, we also saw that the thermal lifetime was longer than a day. So we then tested our compounds for their activity against S-phospholipase inhibitors, and we saw that the thermally adapted sample of azo1 showed very low activity, while irradiation resulted in a more than tenfold activation. And also we showed that after cycles of irradiation with 365 and 420 nanometer light, they showed a reversible inhibition. Nevertheless, we observed an overall 100-fold decrease in activity of azo1 as compared to the parent sulfonamide 1, which might be due to the loss of crucial hydrogen bonds with the oxy anion hole of the target. In the second case, we chose Belinostat, which is an FDA-approved histone deacetylase inhibitor that is used currently in chemotherapy. Docking poses suggested that also here the cis isomer would keep the band geometry, and the subsequent molecular dynamics simulations also revealed that in this case the binding pocket is more solvent exposed and there was more inherent flexibility of the ligands. So this could suggest that the differences we're going to see are going to be less pronounced. Then we photochemically characterized our ligands, and we again did it in an assay buffer. We saw that the compound could be switched with the same wavelengths, also with no fatigue in reducing conditions, and with a PSD of 95%, which is almost quantitative. And again, the cis isomer would be quite long-lived. Finally, we tested our compounds for HDAC inhibition, and we saw that Azo2 overall had potency in the sub-micromolar range. UV radiation resulted in a twofold activation, thereby showing the desired cis-on behavior of the inhibitor. And in this case, we observed a smaller 12-fold decrease in activity when compared to the original ligand. And in this case, the bisulfonamide has a less essential role in binding and does not contribute specific interactions. So to conclude, we show that bisulfonamides are promising cis-oid azosters for photopharmacology because of their bent geometry and favorable dipole moment. But at the same time, the pharmacophoric role of the bisulfonamide influences the outcome of the azologization. And we have a trade-off between losing the original potency and achieving large difference in potency between the photoisomers. So in the first case, we formed the bisulfonamide formed key interactions with buried residues of the protein. So the azoster suffered a larger loss in potency, but also showed a larger difference in potency between the isomers. And on the other, in the other case, we have a sulfonamide that is just a linker to guarantee optimal contacts with the surface of the protein, but it doesn't contribute specific interactions. And in this case, when we substituted with an azobenzene, we observed a smaller loss of original potency, but also a smaller difference in potency between our photoisomers. But overall, I hope I showed you that azologization of cis-oid structures has the potential to guide the rational design of cis-on photoswitchable antibiotics and ligands in general. So now it's time for thanking people. So I would like to thank my supervisors, Ben Feringa and Victor Szymanski, for the great working environment, also the whole group. But I would also like to thank our collaborators at the University of Groningen for molecular dynamic simulations and help with the essays. And finally, Alert PhD program for funding. And of course, I'd like to thank you for your kind attention.
Thank you, Pier Michele, for your presentation, for your contributions, and congratulations for your results.
Thank you.
So before, I would like to check if there are some comments or remarks from the audience. I don't see any. Okay, so I have one comment, more or less one, it's a curiosity actually. I was wondering if you can use sulfonamide as the aryl sulfonamide also to modulate the physicochemical properties of ligands.
So we, that's a very interesting question. That's definitely possible, but there are not, let's say, guidelines yet how to do that. We know that cis-azobenzene has a better solubility than trans-azobenzene in water. So that is already a change we can predict. But then, let's say, specific contributions, there are there's no consensus, I would say. But at the same time, we also observed that the two isomers show a different penetration, for example, in bacteria or cells. So that's still being studied at the moment.
Okay. Another question from Sveva Pelliccia. Two questions, actually.
Yeah.
She congratulates with you, and she said, have you considered the replacement of aromatic rings with, for example, other heteroaromatic rings? And the second question is, what about the penetration of light through tissues?
Yeah. So for the first point, definitely, actually, in the Cambridge structure database search that I did, and also on the PDB search, I allowed for any heterocycle there. But then, of course, this many times when we have an azobenzene with an heterocycle, then we will have, for example, shorter half-lives or different photochemistry. So we chose this standard simple system to prevent any, let's say, side problem that could arise. But that's definitely doable. And also, azopyrazoles are being more and more popular at the moment as photoswitches. So they are used with heterocycles. And For the second point, that's also a long-standing issue in the field. So there are two ways to deal with this problem. One is to develop photoswitchable drugs that can be activated with red or near infrared light, and this has the highest tissue penetration, of course, of the whole spectrum, and that's one way. Or in general, the delivery of light can be achieved with the technology that is developed by photodynamic therapy, and we can insert LEDs in with very thin and non-invasive cables. So that would be doable also deeper in the tissue.
Okay. Okay. So I hope that this answer will satisfy Sveva. In case you can contact each other by private on our chat. So by me, thank you, Pier Michele, for your contribution and for being with us today at the AMYC.
Thank you very much.
So now is the turn of our last oral speaker for the oral communication. Now is the turn of Dr. Carmen Cerchia from the University of Naples Federico II from Italy. Carmen, are you with us?
Yes.
Okay, so she will talk. She will present a talk entitled "Sulfonimide and Amide Derivatives as Novel PPR Alpha Antagonists." So, Carmen, you can start whenever you want and share your screen, and the stage is yours. Welcome.
Okay, so yes, I don't know if you can see me or hear me well.
Yes, we hear you and see you. We also see your screen.
Nice, perfect. So
okay, full screen mode.
Yeah. So first, I'd like to thank the organizing and the scientific committee for giving me the opportunity to present this work that has been carried out in collaboration with the groups of University of Chieti and University of Bari. Oh, well, something went wrong. Sorry. Yes. No.
It's fine. It's fine. We see your screen.
Yeah, but I can't
change the slide.
Yes.
Carmen, let's try to share the PowerPoint, not the desktop.
Yes, I did.
Okay.
But I can't.
Doesn't work. So let's try to close the application and restart it.
Okay. Sorry for this. Just a second. It seems like it can see the PowerPoint for sharing.
So.
It's weird because last day was fine when we.
Okay. So in case, so, but did you try to, Carmen? Okay.
Yes.
But did you try to launch the full screen mode of the presentation and then share it?
Yeah, so, well, I must share the screen and
So yes, now we see your desktop.
Now can you see the presentation?
Okay, now we see also the slide.
Okay.
Yeah, we see the third slide.
Okay, yes.
The background and the second one, and then now the first. Okay, now it's working.
I think it's working
Perfect.
now. Thank you, sorry.
No problem.
So sorry. So yes, I was saying that PPARs are peroxisome proliferator activated receptors. They belong to the nuclear receptor superfamily. There are 3 subtypes, alpha, beta, delta, and gamma, and they are mainly implicated in lipid metabolism and glucose homeostasis. Thus, they are therapeutic target for the treatment of metabolic syndrome. PPARs share a large Y-shaped ligand binding domain in which helix 12 is implicated in the activation of these receptors. The LBD can be divided in a polar arm 1, a hydrophobic arm 2, and an end strands at arm 3. So the correct folding of helix 12 allows the recruitment of coactivators to start the transcriptional process. But if a ligand, for example, an antagonist, interferes with the correct folding of helix 12, the transcriptional process is repressed. Moreover, a balanced activation profile of PPARs is emerging as a novel therapeutic opportunity to counteract metabolic disorders. Thus, partial agonists or even antagonists have been synthesized to explore the beneficial effects of a reduced PPAR activation. In addition, PPARs are emerging as a potential therapeutic targets in the treatment of cancer overexpressing these receptors. For example, PPAR alpha antagonists have shown antitumor effects in different cancer cell lines. As an example, the antagonist JW6471 displayed an anti-proliferative effect in the head and neck paraganglioma cell lines. Thus, building on this information, an agonist to antagonist switch was attempted to pursue the discovery of novel PPAR alpha antagonists. Moreover, it has been already proved that the substitution of the head group, the carboxylic function head group, was a viable strategy to obtain antagonists. Thus, starting from 2 lead compounds reported by the group of the University of Chieti, the stilbene derivative lead compound 1 and the phenyldiazenyl lead compound 2, the carboxylic head group has been replaced with the methyl and the benzyl sulfonamide group, obtaining compounds 1A and 1B and 2A and 2B. These compounds were tested in a transactivation assay, and compound 2B in particular completely inhibited PPAR alpha transcriptional activity. Thus, it was tested it was selected for the design of further derivatives, given its low micromolar IC50. So further derivatives were designed starting from compound 2B, and in addition, the azo moiety at the tail group was replaced with an amide or a urea at the tail group, obtaining compounds 10A2E and 13A2E. The compounds were again tested to evaluate the transcriptional activity, and then they were tested as antagonists in a competitive binding assay using a fixed concentration of reference agonists and increased concentrations of the tested compounds. The results showed that the most potent antagonists were compounds 3A to 3E, 10E, and 13E that were selected to perform gene expression analysis to evaluate their effect on the expression of a PPAR alpha target gene. In fact, CPT1A is an enzyme involved in fatty acid beta oxidation. And it was notable that the combination of an agonist GW7647 with the tested compound was able to significantly repress CPT1A expression, thus further supporting the antagonist behavior of these compounds. Next, we evaluated the anti-proliferative activity of these compounds on 8 cancer cell lines representative of 4 cancer types: pancreatic, colorectal, paraganglioma, and renal. And the results show that the compounds 3C, 3D, and 10E displayed the most consistent anti-proliferative activity among the 8 cancer cell lines, and with a more marked effect on paraganglioma cells. Thus, the 3 compounds were tested in dose response MTT assay on paraganglioma cell lines, and the results show that 3C, 3D, and 10E were able to reduce the cell viability of paraganglioma cell lines in a concentration-dependent manner without affecting the normal fibroblasts. The median CC50 values were also in the low micromolar range. The most potent compounds displaying the lower IC50 values were selected for docking studies. In particular, the results of the docking of compound 3A displayed that the benzyl sulfonamide moiety is able to engage an H bond with the tyrosine 314, and tyrosine 314 in turn engages a paste-taking interaction with the head group of this compound. Compound 10E also engaged an H bond with the tyrosine 314, and the tail group formed 2 additional H bonds, stabilizing the beta sheet and helix 3. The overlay of the docked poses of 3A and 10E with the crystallographic pose of the antagonist GW6471 displayed a very similar binding mode. Moreover, the head groups of 3A and 10E are projected in an area that is usually occupied by the residue of tyrosine 464 in the PPAR-alpha agonist bound conformation. In fact, this residue is critical for the receptor activation. In this case, the 2 compounds do not interact with this residue, likely because of steric clashes, and so helix 12 is forced in a conformation that is not able anymore to recruit the coactivators, and thus the transcriptional process is repressed. Docking studies allowed deriving some clues about the structure-activity relationships. In particular, the substituents at the para position of the phenylsulfonamide group were explored, and it turned out that only lipophilic and quite small substituents are tolerated at this position. In fact, the introduction of a bulkier benzyl amide group was not tolerated because it increased the IC50. And the docked pose of the 3G displayed that in this case, the head group is completely shifted upward in arm 1, so that the H bond with the tyrosine 314 is lost. As regards compounds for A to D, the presence of the amide head group decreased activity. Also for compounds 10A to E, the presence of the phenylsulfonamide head group was critical for the antagonist activity. In fact, in this series, only compound 10A displayed IC50 in the low micromolar range. The normal beauty substituent in compound 10B was not able to interact anymore with the residue of tyrosine 314, but instead was shifted toward the glutamine 277, thus accounting for the slight receptor activation at the low or inactive antagonist activity. Finally, compound 13E displayed a very similar binding mode to compound 10E. Moreover, the presence of the urea moiety increased the affinity and the activity, probably because of the possibility of this tail group to interact with the residue of cysteine 275 on helix 3. So in conclusion, a novel series of sulfonamide and amide derivatives were identified as novel PPAR-alpha antagonists, and among them, compounds 3C, 3D, and 10E displayed remarkable anti-proliferative effects in 2 paraganglioma cell lines, and molecular modeling studies allowed to describe the binding mode of this compound and to help the elucidation of structure-activity relationships. This work has been published in a special issue in ACS Medicinal Chemistry Letters last year. And finally, I'd like to thank all the people involved in this project, Professor Lavecchia, the group of Professor Loiodice at the University of Bari, and the group of Professor Amoroso at the University of Chieti. And thank you for the attention.
For your presentation, for being right on time. I'm going to check if there are some. I don't know. Maybe there is one.
Stefano, I have a question.
From Francesco Di Leva.
Yeah, Carmen, can you hear me?
Yes.
Okay. Hi. So I thank you for the nice presentation. So since in general nuclear receptors share similar structural requisites for ligand binding, I was wondering if you also checked the selectivity of these compounds against other receptor of the same family. I don't know, the vitamin D receptor or the retinoic acid receptor or something like that.
Yeah, actually, the only other receptor we checked was PPAR gamma. In fact, compound 2B displayed a slight inhibition also on PPAR gamma. However, we didn't check on the other nuclear receptors, the agonist or antagonist activity.
Okay, I have another curiosity. So based on your work, I was wondering if you conclude that the different affinity and of course activity profiles of your compounds are linked more to the bulkier, so the bulkiness of the tail substituent, or might depend also on their, I don't know, chemical physical properties, because you have a sulfonamide or substituent of different natures. So which is the so if in your opinion the bulkiness or the physical chemical properties have the greatest importance in determining the activity of your compounds?
Yes, the bulkiness of the head group was critical. In fact, there was a really we saw that the bulkier substituents were basically much less active. But also the tail group has a role. In fact, the urea group at the tail group was a very good compromise.
Okay, so using both, it might depend on both.
It's a yeah, most probably it's a combination, but the bulkiness of the head group was critical.
Okay, thank you.
Thank you. I stopped sharing the screen and thank you.
So, thank you, Carmen, for your contribution and for your presentation once again. So now I think we are entering the last part of our morning session of the medicinal chemistry session. And so we are heading to 3 short communications, slide and talk. The first participant will be Dr. Lorenzo Baldini from the University of Florence, Firenze in Italy. Are you with us, Lorenzo? Yes, now I see you. Lorenzo will present a talk entitled Design of Dual Peptidomimetic Gelatinase Integrin Inhibitors for Tumor Angiogenesis Targeting. Lorenzo, welcome, and the stage is yours. You can start. Thank you.
Thank you. Okay, I'm sharing the presentation. Can you hear me and see it?
Yes.
Probably? Okay. So,
but we don't see. But sorry, sorry for interrupting you. But we don't see your screen. We just see your
all
face.
white.
No, we don't see your screen.
Ah, sorry. Maybe now.
Yes, but not in full screen mode.
Okay,
so you need to.
Okay.
Okay, now it's perfect.
Okay, sorry.
Thank you.
Okay, good morning to everyone. So I introduce you my work, which is entitled Design of Dual Peptidomimetic Gelatinase Integrin Inhibitors for Tumor Angiogenesis Targeting. So tumor angiogenesis is an essential step in metastasis in which the tumor mass product in other organs through the creation of new blood vessels. The gelatinous subfamily of matrix metalloproteases and integrins are deeply involved in this process due to their activities. Indeed, the first are able to degrade and remodel the extracellular matrix due to their proteolytic activity, while the integrins, which activate themselves the gelatinases, take part in processes like cellular migration and adhesion. And for this, they are both important target for metastasis. On the basis of reported integrin inhibitors, we synthesized analogous molecules which have been variously functionalized with a basic functional group at the end of a propylic chain, which will interact with an acidic amino acid residue in the active sites. Then a chelating functional group, which can be a carboxylic or hydroxamic acid functional group, and then an alpha arysulfonamido functional group, which will interact with hydrophobic pockets into the active sites. So 5 compounds have been synthesized and tested through cellular and inhibition tests toward alpha V beta 3 integrin and MMP2 respectively. And here in the table are reported the results obtained for these 5 compounds. In order to rationalize ligand-protein interactions, molecular docking analysis has been carried out. And here is reported a representative pose of N33 into the active site of MMP2, in which we can visualize the presence of different interactions, starting from a salt bridge between the basic amino functional group with glutamic acid 210, chelating interaction between the zinc metal ion in the Midas region and the carboxylic acid functional group, then 2 hydrogen bonds due to the presence of the sulfonamido functional group, and then hydrophobic interactions due to the presence of the biphenyl structural motif with the amino acid residues in the S1 first pocket of MMP2. So I can conclude that hydroxamic acid is a good functional group for the inhibition of both MMP2 and alpha V beta 3 integrin. Then the basic amino functional group is demonstrated to be more effective on MMP2, while the guanidino functional group has been more effective on alpha V beta 3 integrin. Then N33 has demonstrated to be the only molecule with carboxylic acid functional group that showed inhibitory activity toward MMP2. And at the end, the entry 2 has demonstrated to possess potentialities as a dual gelatinase integrin inhibitor. And so at the end, I thank you for your kind attention. Thank you.
Thank you, Lorenzo. Thank you for your presentation or your contribution.
Thank you.
Now is the turn of Sara Lamanna from the University of Naples Federico II. I don't know if she's already with us. Here it is. Here we are. Good morning. She will present a talk, a slide talk presentation entitled New SOX Cyclic Peptidomimetics from Engineering to Functional Evaluation.
Do you see my presentation?
Yes.
Okay.
You can start.
Good morning. As already said, today I'll talk about the discovery of new peptidomimetics of SOX1 protein with a potential application in cardiovascular diseases. SOX1 protein is a negative feedback regulator of the JAK-STAT pathway, a pathway essential for the regulation of a wide range of cytokines and growth factors, whose dysregulation has been linked to several cardiovascular diseases, including atherosclerosis. Thus, the identification of SOX1 mimetics appears to be a good starting point to develop potential therapeutics. SOX1 protein has a modular structure and in particular present a kinase inhibitor region that is required for the high affinity binding to JAKs and for the inhibition of the JAK kinase activity. In the past, several studies outlined the anti-inflammatory properties of a peptide covering the key region. And in my research group, to identify new peptides that are able to mimic the key activity, they screened a focus simplified peptide library, identifying a linear peptide named PS5 that was able to act as a ligand of JAK2 catalytic site with a KD in a low micromolar range. Unfortunately, this peptide revealed to be too much flexible. So to rigidify the structure and to improve the stability of PS5 peptide, following a medicinal chemistry approach, we recently designed smaller side cycles, internal cyclic PS5 and internal cyclic PS5 null1. And as shown, both peptides are formed of only 10 amino acids, and in the cycle is achieved by a lactam bridge between side chain of aspartic acid and lysine. And in internal cyclic PS5 null1, to investigate the role of the phenylalanine in position 58, that results a crucial residue for the interaction with JAK2, we decided to substitute this phenylalanine with a non-natural naphthylalanine. First of all, we performed in vitro assays, and to evaluate the conformational properties, we performed NMR studies that indicated that both peptides are much more rigid, as expected, with linear PS5. And from a functional point of view, we performed the microscale thermophoresis experiments that indicated that both peptides were able to bind to JAK2 catalytic site with a KD in a low micromolar range, and in particular, the substitution of the phenylalanine with naphthylalanine increased the binding ability. From a cellular point of view, we tested these peptides in vascular smooth muscle cells. And first of all, we checked their ability to impair the cell migration. That is an important process involved in the atherosclerotic plaque formation. And as shown in the wound healing assay, a combination of pro-inflammatory cytokines, interferon and interleukin-6, promoted a time-dependent increase of cell migration, while both peptides show anti-migratory effect. Immunofluorescence experiments revealed their capability to inhibit both STAT1 phosphorylation and consequent nuclear translocation. And moreover, they significantly reduced the mRNA expression of STAT-regulated pro-inflammatory genes, such as CCL2, CCL5, and CXCL10. And interestingly, both peptides reveal also antioxidant effects. And in particular, we analyzed the production of superoxide anion in both vascular smooth muscle cells and macrophages. by staining cells with dihydroethium, that is a dye able to cross cell membrane and produce fluorescence upon reactive oxygen species oxidation. And as shown, the fluorescence induced by a combination of pro-inflammatory cytokines was drastically reduced in presence of both analogs. In conclusion, in these studies, we developed the novel constrained peptidomimetics of the kinase inhibitor region of SOX1 protein, able to act in a cellular context as mimetics of the entire SOX1 protein. And this data indicate that mimetics of SOX1 could be excellent agents for cardiovascular diseases. Thank you for your attention.
Thank you, Sarah. Thank you, Sarah, for your presentation. And now is the turn of Diego Garcia Jimenez from the University of Turin, Italy. He will talk about a presentation about PROTACs, solubility, and computed polarity. Thank you. Welcome, Diego, and feel free to start.
Good morning to everyone. Can you hear me?
Yes. We also see your screen, okay. So just full screen mode, and then it's fine.
So good morning to everyone. My name is Diego Garcia Jimenez. I'm a PhD student of the University of Turin, and today I will be presenting our latest work named PROTACs, Solubility and Computed Polarity. First of all, I'd like to introduce PROTACs as beyond the rule of 5 molecules, displaying large and flexible structures that make them responsible for low solubility and viability restrictions. Moreover, since PROTACs are more or less a recent technology, there is a lag in terms of permeability solubility data that made us think of an experimental design regarding solubility. That's why in this recent work, we tried to determine experimental thermodynamic solubility. This is the solubility when the thermodynamic equilibrium has been reached. We also wanted to correlate experimental solubility values with molecular descriptors of the structure, in this case polarity. And finally, the third objective was to study if the 3D conformations of these PROTACs, as I have explained, they're flexible and very complex, to see if the 3D structures have a major role in the prediction of solubility. To do this, the job we performed was the creation of a PROTAC chemical space, taking advantage of the PROTAC database that was released at the end of last year, that includes more than 2,600 PROTACs published already. We took advantage of this database and we calculated some descriptors, such as number of carbons, meaning as the size descriptor, phi as a flexibility descriptor, and TPSA, the total polar surface area, as a polarity descriptor. With these 3 descriptors, we were able to identify in the total chemical space different regions with privileged pharmacokinetic properties. Moreover, in this chemical space, we identified commercial PROTACs that were available. We bought these PROTACs and we performed an experimental assay named the shake flask method. This method consists on the suspension of 1 milligram of the PROTAC in 10 millimolar PBS at pH 7. Then the samples were stirred at 25 degrees for 1 hour at 500 revolutions per minute. And then finally, samples were filtered and finally quantified by HPLC UV. The goal of this part was to obtain accurate solubility values for the 21 PROTACs. Once we obtained these values, the next step was to see if the different solubility values had a relation with the chemical structure. Therefore, we tried to correlate these solubility values with polarity and lipophilicity descriptors. First of all, we start with 2D descriptors that are not dependent on the 3D conformation. And we first started with TPSA, which is a fragment-based descriptor that takes into account the maximum polarity that a structure could expose, considering only 2D aspects. We found that a very good correlation, 0.42, was obtained, and also for lipophilicity values, we used the Marvin software, and we obtained a 0.7 correlation, which was very promising. Up to this point, we can say that solubility can be easily modeled only with polarity and lipophilicity 2D descriptors, which is already a great result. Moreover, we wanted to go one step ahead, and we tried to use 3D structure properties. Therefore, we used the software Maestro to perform a conformational sampling, studying the different conformations that each product could adopt in water. In this graph, we can see in the x-axis the 3D PSA of the different conformations, and in the y-axis the experimental log S we had already found. For this case, we saw that the different regions of the conformational sampling did not improve the correlation found with TPSA. We found 0.38 for minimum, all the way to 0.37 for the mean. Finally, we can conclude for our work that polarity and lipophilicity are moderately correlated to solubility, as I have just explained. Also, that solubility does not depend on the 3D conformation. And finally, that products exist as an equilibrium of conformers in water, since minimum or high maximum conformations do not change the correlation with solubility. Finally, I would like to say that at the moment we're working on the development of a solubility model that doesn't need the measurement of experimental values, just with computed values. This could be actually a breakthrough, because it would mean not wasting some product quantity, which is actually very valuable. Thank you very much for the time of this presentation, and I would be happy to answer to any questions in my email. Thank you very much for your time.
Thank you, Diego, for your presentation. You were the last one of this morning's session. I hope that some people will contact you personally by email to share some ideas or comments. For today, we are closing the morning session of the second day of our AMYC-BIOMED 2021. I would like to personally thank all the speakers, the invited lecturer, Professor Kathleen, Dr. Neil London from the Weizmann Institute for his invited talk about new covalent inhibitors, and all the other speakers of today, and of course all the attendants of our session. So now we are just 10 minutes in delay, and we will continue with the afternoon session around about 2:30, and so keep on to be with us, and see you later. Now we're gonna stop for the lunch